So, Dr. Xu, actually, I'm gonna first start with the agenda briefly and go to session 1. Thank you, Dr. Yang, for the nice introduction, which brings great excitement for us to kick off the session. And thank you, Dr. Liang, for introducing HMS-CSSA. Today, we're going to have 3 sessions, 2 in the morning and 1 in the afternoon, covering exciting sciences from immunity, neuroscience to structural biology. There will be a lunch break after session 2, and we will have a ceremony for this year's recipients of HCLS Distinguished Research Award. Please stay tuned for that. So I would like to remind the audience that please do not record on your own. Each speaker would have 25 minutes for talks, 5 minutes for Q&A. Please type your question in the Q&A box only, and our moderator for each session will pick questions from there. So we will make the chat box available during the breaks. Okay, that's for the agenda, and now let's get started with first session, immune tolerance. There will be 3 speakers, Dr. Arlene Sharpe as our keynote speaker, followed by Dr. Chen Dong and Dr. David Haffler. Dr. Xu Zhou is our moderator for session 1 today. So let me introduce Dr. Xu Zhou briefly. Dr. Xu Zhou is assistant professor of pediatrics at Boston Children's Hospital and Harvard Medical School. He went to Peking University and he obtained a PhD at Harvard and did his postdoc training at Yale. Since 2021, he established his own lab to study how immune cells communicate with their neighbors and surrounding environment to control tissue homeostasis and inflammation. Dr. Xu Zhou, thank you. You may now turn on your audio.
Thank you for the introduction for the session. So I think this community is very familiar with our first speaker, Arlene Sharpe. Arlene is the George Thapman Professor of Comparative Pathology and the founding chair of the Department of Immunology at Harvard Medical School. Arlene also has several academic affiliation and leadership positions all around Boston institutions. Particularly, she led the Cancer Immunology Program at Dana-Farber. She's a co-director of the Evergrande Center for Immunological Disease. She also co-led the Massachusetts Consortium of Pathogen Readiness in response to COVID pandemics. Arlene earned her MD and PhD degrees from Harvard Medical School, and then she completed her residency just next door at Brigham Women's Hospital. And then Arlene stayed around the Longwood area to start her own independent career. Her lab has discovered numerous great findings to the community, including discovery and elucidation of the function for T-cell co-stimulatory pathway, including CTLA-4 and Pd1 pathway, which we believe we'll hear more about today. Her lab currently focuses on the role of T-cell co-stimulation in regulating T-cell tolerance, antimicrobial, and anti-immunity. Dr. Arlene received numerous awards for her seminal contribution, such as the William Coley Award in Tumor Immunology, the Warren Albert Foundation Prize, the Smiley Award from Society for Immunotherapy for Cancer, and also the Lifetime Achievement Award from the American Association of Immunologists. Dr. Arlene is an elected member for the National Academy of Science, the National Academy of Medicine, a fellow with American Association for Cancer Research, and the National Academy of Inventors. In addition to being an exceptional scientist and leader, Arlene is also a role model as a mentor for undergraduate students, graduate students, postdocs, as well as many junior faculties. Arlene, thank you for being here with us today. I look forward to your talk.
Well, thank you so much for that very generous introduction. I'm really delighted to be here this morning and honored to be one of your speakers today. I'm delighted to have the opportunity to share some of the work from my lab. I'll start by sharing my screen here. And can you see my slides?
Yes.
Okay, excellent. Very good. So in my talk this morning, I'm going to focus on the Pd1 pathway from 2 perspectives. Understanding this pathway deeply, I'll first give a brief introduction to the pathway and discuss some of its functions, and then share with you some of the work in my lab where we've developed an in vivo gene editing platform that allows us to discover and validate new cancer immunotherapy targets. These are my disclosures related to the Pd1 pathway. So I know this is an audience that's a very broad audience, so I'll start with a few general introductory remarks. So immunology has offered hope for curing cancer for over 100 years. But what is different now is the strategy. We're beginning to appreciate that the tumor microenvironment has many layers of immunosuppression. Tumors evolve to evade immune attack. And one promising strategy that now is being used is to block pathways that are being used by tumors to inhibit anti-tumor immunity. This approach is known as checkpoint blockade. And essentially this approach, the goal of it is to stop the tumor from turning off the immune system. So what are these pathways? These are key immunoregulatory pathways that regulate T cell responses. For T cells to become activated, they need to receive two signals. The first signal confers specificity to an immune response and involves antigen recognition. Antigenic peptide is presented on MHC molecules to the T cell receptor CD3 complex. But in order for complete activation to occur, the T cell also needs to receive a second signal known as the co-stimulatory signal. And when the T cell receives both of these signals, it can become activated, and then the cells proliferate and can differentiate into effector cells. The CD28 receptor provides these positive signals that allow a naive T cell to become activated and differentiate into effector cells. But there are also negative second signals that can attenuate or reduce T cell responses. And the pathway I'm going to focus on this morning, the Pd1 pathway, provides one of these very important negative second signals. And I'll use this slide to introduce the Pd1 pathway. The Pd1 receptor is upregulated on T cells upon their activation. And when Pd1 is engaged by either of its ligands, PDL1 or PDL2, it can become phosphorylated on tyrosine motifs in its cytoplasmic tail. This can lead to the association of protein tyrosine phosphatases such as SHP2, and then dephosphorylation of kinases downstream of the T cell receptor and CD28. As a result, there is reduced signaling through the T cell receptor and CD28 and reduced T cell responses. For example, reduced cytokine production and reduced target cell lysis. PD1 has two ligands, as I mentioned, PDL1 and PDL2, and the expression patterns of these ligands are summarized on this slide. One of the things that really fascinated me from the early days working together with Gordon Freeman's lab at the Dana-Farber Cancer Institute, we discovered that PDL1 and PDL2 could bind to PD1, thereby defining this pathway in 1999 and 2000. And one of the things that really fascinated me from the beginning were the expression patterns of these ligands, in particular PDL1 is shown on this slide. PDL1 can be expressed on a variety of hematopoietic cells, including dendritic cells, macrophages, B cells, and T cells. But in addition to hematopoietic cells, PDL1 can be expressed on a variety of types of non-hematopoietic cells, including vascular endothelial cells, a variety of types of epithelia, cells in the liver, cells in the islets of the pancreas, syncytial trophoblasts in the placenta, and cells in the eye. This expression pattern suggested to us that PDL1 may not only control responses in lymphoid organs, but also in a variety of tissues and locally regulate T cell responses. In contrast to the broad expression pattern of PDL1, PDL2 is expressed primarily in hematopoietic cells. As summarized in this slide, it can be expressed on dendritic cells, macrophages, B cells, and some T cells. It can be expressed on a few types of non-hematopoietic cells, in particular in the lung, on airway epithelia. Interferons, alpha, beta, and gamma are potent stimuli for upregulating PD-L1 expression. And PD-L2 can also be upregulated by interferons, but IL-4 and GM-CSF are the more potent stimuli. While PD-L1 and PD-L2 share PD-1 as a binding partner, each of these ligands has a second unique binding partner. Working together with Gordon Freeman's lab, we showed that PD-L1 could also interact with B7-1 or CD80. Using selective antibodies that blocked this PD-L1 B7-1 interaction, we showed that this was also an inhibitory interaction, and that this pathway could control and limit effector T cell responses. PD-L2 can interact with repulsive guidance molecule B or RGMB, and this interaction has an important role in controlling tolerance, particularly in the lung microenvironment. When I think about the PD-1 pathway, it's really a paradigm for why it is we have inhibitory signals in the immune system. And I've summarized on this slide some of the functions of this pathway. The PD-1 pathway serves to counterbalance positive signals through the T cell receptor, as well as co-stimulatory receptors, including CD28 and CD226. Inhibitory signals through this pathway have an important role in mediating and maintaining T cell tolerance and protecting autoimmunity. In addition, as I mentioned previously, the expression of PD-L1 on tissues fascinated me and led us to do further experiments to probe the function of PD-L1 on tissues, and first using bone marrow chimera approaches and later conditional knockout approaches, we showed that PD-L1 on non-hematopoietic cells can control resolution of inflammation and also protect target organs from autoimmune attack. So, for example, self-reactive CD4 and CD8 cells are restrained from damaging target tissues through the PD-L1 expression on target organs, such as the islet cells in the pancreas. In addition, more recently, we found that these inhibitory signals can also control cell fate decisions, and have shown in work that we published last year that signals through this pathway can promote optimal differentiation of CD8 T cell memory, that if we lack this pathway, we end up with short-lived rather than long-lived memory cells. But it's the smartest immunologists, the tumors and the microbes, that over and over again have exploited this pathway to their own advantage. This pathway contributes to T cell dysfunction, or known as T cell exhaustion, that can develop during chronic infection and cancer. This is a state that's defined by poor effector functions, so poor ability of these T cells to produce cytokines or to kill cells that are infected by microbes or tumors. There's sustained expression of inhibitory receptors like PD-1, and we now know that this is a unique transcriptional and epigenetic state. Since I know that there are people here from a variety of scientific backgrounds, I'll just take a moment to explain T cell exhaustion and here compare an acute infection to a chronic infection, which is where the role of this pathway in T cell exhaustion was first appreciated. So there are some viruses like the influenza virus that infect cells, and this can lead to the activation of a naive CD8 T cell and result in its differentiation to an effector cell that then can clear the virus. Some of these cells go on to become memory cells, so the next time one encounters this virus, you're poised to respond and have cells that can rapidly produce cytokines and kill. This is the basis for vaccination. So we get cells that are flu specific, for example, and the next time you encounter these through an infection, you're poised to respond. However, there are some viruses that can cause a chronic or persistent infection, such as hepatitis B, hepatitis C, and HIV. The gold standard mouse model that's used to study this is a model of lymphocytic choriomeningitis virus known as LCMV. And in addition, we've come to appreciate that in cancer as well, these exhausted T cells exist. What these share is that there is chronic antigen persistence, and as a result of persistent antigen stimulation of T cells, these cells enter into an exhausted state where they progressively lose functions. First, they lose the ability to make IL-2, then they lose the ability to make other effector cytokines and have poor ability to kill, and some of these cells can just fade away. We now appreciate that there's heterogeneity in these exhausted T cell subsets, some that are more progenitor-like and some that are fully dysfunctional over time. The role of the Pd1 pathway in T cell exhaustion was first discovered in the LCMV chronic infection model. And this is work that is part of a long-term collaboration with Rafi Ahmed, John Wary, Dan Barber, and Gordon Freeman. And some of the early work was inspired by work that had gone on in Rafi Ahmed's lab using transcriptional profiling. And in these days, it was really early work in microarrays that showed that PD1 expression differed during acute and chronic LCMV infection. There's an acute form of LCMV where the virus is cleared, and then a chronic form where the virus persists. And what you're looking at on this slide is looking at the expression of PD1 during an acute infection and comparing that to a chronic infection. And what you can see here is that during an acute infection, when there's virus and TCR stimulation, PD1 expression goes up, but as the virus is cleared, PD1 expression goes back to baseline. In contrast, during a chronic infection, PD1 goes up and stays high because of the chronic infection that ensues. Work in collaboration with Rafi's lab showed that the cells that highly express PD1 are exhausted, and these cells are less functional. And importantly, when the PD1 pathway was blocked using anti-PD-L1 antibodies, this reduced the viral burden and reinvigorated these exhausted cells so they could now respond to the virus and reduce viral burden and kill these infected cells. What the slide also shows you is that PD1 can be upregulated on recently activated cells as well as exhausted cells. So one cannot determine by PD1 expression alone whether a cell has become recently activated or is exhausted. Further markers are needed. And so these exhausted cells, as I mentioned, are not only expressed during chronic viral infection, but they also can be expressed in the setting of cancer. But one of the special features of cancer is that the tumor cells themselves can express the ligands for PD1. And what I'm showing you here on the left is a kidney tumor, and on the right is a non-small cell lung cancer. And we're using immunohistochemistry to visualize PD-L1 expression on the tumor cells. And this brown staining is showing you PD-L1 expression ringing these tumor cells on the left and on the right. And this is work done in collaboration with Gordon Freeman's lab, who developed these anti-PD-L1 antibodies, and in collaboration with Scott Rodig and Sabina Signoretti and David McDermott, with whom I collaborate on kidney cancer work. And so we now appreciate that PD-L1 can be expressed on the surface of about a third of solid tumors and certain hematologic malignancies. And work from our lab and Bob Schreiber's lab has shown that PD-L1 expression on the tumor cells themselves can contribute to inhibition of anti-tumor immunity. So in the setting of cancer then, what we have are T cells, CD8 T cells that highly express PD1, and these are dysfunctional. PD-L1 can be expressed on the tumor cells themselves, on hematopoietic cells in the tumor microenvironment, and as well as non-hematopoietic cells, including the vasculature and fibroblasts. And the antibody drugs that have been developed, antibodies to PD1 or PD-L1, can block this interaction and unleash a potent anti-tumor immune response, resulting in increased cytokine production and increased killing. We now know that PD-1 blockade can act on T cells in the periphery as well as in the tumor. And so CD8 T cell responses are spatially dynamic. They can be initiated in the tumor draining lymph node, and then these T cells can traffic to the tumor where they can execute their effector functions and kill these tumor cells. Recent work has shown that the tumor draining lymph node contains a reservoir of less dysfunctional tumor-specific T cells, and these cells can be mobilized to fight the tumor, and that the PD-1 inhibitors can act on T cells in the draining lymph node. And we're beginning to appreciate the trafficking of new clones from the lymph node into the tumor as an important mechanism of anti-PD-1 induced anti-tumor immunity, and this has been appreciated over the past several years. And so, as you know, this has been translated to therapy, block this inhibitory pathway to unleash potent anti-tumor immune responses. And there are now antibodies to both PD-1 and PD-L1 that have been approved for treatment of patients with cancer. And I've listed here some of these antibodies to PD-1 and PD-L1 that have been approved for cancer therapy. However, as shown on this slide, while there's broad anti-tumor efficacy of these PD-1 pathway inhibitor, only a subset of patients respond. What we're looking at here of a variety of tumors listed on the left and overall response rates on the right. And you can see that there is a variety of response rates that have a range of responses going from 20 to about 87%. And there's a lot of work going on now to understand mechanisms of response and resistance. And this success has really stimulated incredible creativity and innovation in the areas of cancer immunology and immunotherapy. And so there's great interest in trying to understand mechanisms of resistance and how we can increase this benefit. And what I just want to illustrate here is, in addition to looking at types of tumors, we also know that this inhibition of this pathway has been approved for a class of tumors that are mismatch repair deficient and a variety of different tumor cell types. And one mechanism that we appreciate for the high response rate in Hodgkin's lymphoma is that in this tumor, work from Margaret Shipp at the Dana-Farber Cancer Institute showed that in Hodgkin's lymphoma that there's chromosomal amplification of PD-L1 and PD-L2 and also JAK2, which can then lead to further amplification of PD-L1 and PD-L2. And when together with Philippe Armand, they did a clinical trial in Hodgkin lymphoma, they had an 87% response rate. And so there's great interest, as I mentioned, to benefit this response. And so to understand further, we've done some work to use the TCR as a molecular barcode to investigate exhausted T cells. And I'm summarizing on this slide some work that we published last year, and our work was back-to-back in the Journal of Experimental Medicine with David Haffler, another of our speakers in this session this morning. And we recently published a review on the TCR as a molecular barcode that can further talk about using the TCR to investigate T cell responses. Essentially, what we did in the work that I'm summarizing here is to look both in mouse tumor models and in mouse melanoma patients. And this is work with Mike Rosenblum and Adil Dodd at UCSF, where what we did was isolate blood and tumor cells And then we used the T cell receptor as a way to identify specific antigen-specific T cells. And we compared these tumor-specific T cells in the blood as well as in the tumor, and did paired single-cell RNA-seq and TCR-seq, and looked at the properties of these cells. And in both our studies in mouse models, as well as patient samples, we found that tumor matching cells in the blood were less dysfunctional than their matching counterparts in the tumor. And so it may well be that the blood can provide a window into the ongoing T cell response in the tumor as a result of therapeutic modulation. And so really the clinical utility of this approach is nascent, and it remains to be seen if clonal analyses can be implemented for clinical testing. But these studies are already beginning to show benefits in understanding mechanisms of T cell responses in tumors. And so one of the key questions is, can changes in the tumor matching population in the blood over time indicate responses to ongoing cancer immunotherapies? And there's been some work showing that there can be correlation between highly expanded circulating clones that are Ki-67 positive T cells and response to melanoma patients who have been receiving anti-PD1 or anti-PD1 plus anti-CTLA4. And in contrast, if there's persistence of tumor active T cell clones in the blood that have transcriptional features of exhaustion, this has been correlated with melanoma progression in patients who are receiving anti-PD1. And so really the future is in combination therapy, and the future is now. And I've listed on this slide some of the different approaches that are underway to combine with PD1. And what you can see here is that PD1 is really a building block for all of these different approaches. And I've also listed in red therapies that are in combination that have been approved by the FDA for cancer. So one approach is to combine PD1 with other inhibitory receptors. Exhausted T cells, in addition to expressing PD1, can express other inhibitory receptors, including LAG3, TIM3, TIGIT, and CTLA4. And what has been found is that if you combine these receptors, one can get a better response than blocking PD1 alone. And so this was first seen in mouse models. And now there are a number of clinical trials underway to look at combinations, and the combination of PD1 and CTLA4, as well as the combination of PD1 and LAG3 have been approved for some human tumors, and other clinical trials are underway. Another approach is to block PD1 and then give a kinase inhibitor or some chemotherapies. And some of these combinations with PD1 have been approved as well, and many combinations are underway. And we really need to understand how these combination therapies, how kinase inhibitors and chemotherapies will affect immune cells so one can rationally combine them. Likewise, for immunotherapies, while we know that these cells can express many cell types, many different immunoregulatory inhibitors, we really don't know if different immune cell types or cell circuits are impacted in the same way or different ways. And this is some of the ongoing work in my lab. Other approaches are to block PD1 and give an angiogenesis inhibitor, and this has also been approved, or other approaches, TGF-beta blockade, radiation, epigenetic inhibitors. I should point out that PD1 blockade does not affect the epigenetic landscape of exhausted T cells. And so there's great interest in blocking PD1 along with epigenetic inhibitors. Other approaches are to block PD1 and then stimulate the immune system in some way, giving agonistic antibodies to OX40 or 4-1BB, known as CD137, to stimulate the immune system using different forms of IL-2, toll-like receptor ligands, STING modulators, or certain bispecifics. There's a variety of very clever engineered bispecific antibodies that are now being tested in the clinic. Other approaches are to block PD-1 and then give a cancer vaccine or an oncolytic virus or engineered T cells. These are especially important in certain microenvironments called immune deserts, where there are not an immune cells or T cells in that environment, and one wants to draw these cells into that environment. Other approaches are to block PD-1 and give certain components of the microbiota or certain nutrients. And so really there is a critical need to rationally identify and prioritize targets for combination with PD-1, because there are many more potential targets or approaches than there are patients to test them. And so one approach that we have taken is an approach that is a CRISPR-based screening approach that I'd like to share with you now for the last part of my talk, which we've used to discover new therapy targets and validate them. And this is work led by Martin Lafleur, a very talented postdoctoral fellow in the lab. So we're living in a time where we have a variety of different omics approaches, a variety of single cell transcriptomic approaches, epigenetics approaches, metabolomics approaches that are leading to immense catalogs of genes and pathways that may play a role in regulate innate and adaptive immune responses. So simply identifying these components is no longer the stumbling block, but we need to ascribe functions to each of these genes and pathways. And functional annotation is lagged behind this description. And so what we've done is to develop an approach that allows us to overcome some of these challenges, because one of the challenges in the immune system is that in order to deliver these vectors for gene editing, one has often had to activate these cells such as T cells, and this can perturb T cell states and obscure important immune pathways. So we've developed a lentiviral system that I'm summarizing on this slide, where we've isolated Cas9 expressing stem cells, transduced them with a lentiviral vector that has a reporter, a violet excited form of GFP known as VEX, which is a very good marker, and also importantly, for reasons we don't understand, it's not immunogenic. We then transfer these transduced LSK cells into irradiated recipients to create bone marrow chimeras. And then after immune reconstitution, we can study the chimeras themselves or transfer immune cells such as T cells to new hosts and perturb the system in some way, such as implanting tumors or infecting the animals. This is a versatile approach. We can knock out cells in multiple immune lineages shown here. In the light gray, it's control. The dark gray is a guide of interest. CD20 in B cells, CD64 in macrophages, DEC205 in dendritic cells, PD1, PDCD1 in T cells. And we get very effective knockout. Working together with John Dench and using his algorithms, we've done extensive single cell RNA-seq profiling of control guide transduced cells to show that these cells look basically like naive cells, and their development state is not altered. And when we use TIDE, we've confirmed that there's sufficient gene knockout and minimal off-target editing. So our approach has really been to look at T cell screens, and the approach that we've used is to either a dropout or enrichment, where we have a library of guides here, and we're looking at either enrichment of cells carrying these guides or dropout after selection by infection or tumors. And what I'd like to share with you just to illustrate this system is the screen that we've done T cell exhaustion, looking at a variety of categories here, T cell receptor signaling, knocking out some of our favorite co-stimulatory or co-inhibitory molecules, genes involved in cytokine or inflammation sensing or metabolism. And we've used the LCMV clone 13 model for T cell exhaustion, where we took a library of guides, transduced them into our stem cells, and after immune reconstitution, sorted out the TCR transgenic cells. We have our Cas9 cells that are TCR transgenic that recognize a component of LCMV. And after we transfer these cells into naive hosts, we then infect the animals with LCMV clone 13. And then a week later, we look for dropout or enrichment. And here what we found were genes that were involved in T cell signaling dropout, ZAP70 downstream of the T cell receptor, or CD28. And then some genes were enriched, such as PD1, PDCD1, as well as A2AR and KDR and PTPN2. And I'd like to, in the last few minutes, just show you how we use this system to validate a gene. We first focused on PTPN2 because it's a phosphatase that serves as a negative regulator of T cell receptor and cytokine signaling. It's been associated with tolerance and autoimmunity, and tumors are very clever. They hijack tolerance mechanisms. And in addition, this PTPN2 had been found to have a role in tumor cells. So we thought it'd be very interesting if it, in the same way, if we modulate it, if it promotes T cell function and makes tumor cells more vulnerable to apoptosis. So what we did here was first to validate this by doing a competitive transfer. We had cells that were a 50-50 mix where we knocked out PTPN2 or a control guide and implanted tumors. And we found that the cells that lacked PTPN2 were expanded and that they were better producers of granzyme B. And importantly, when we made bone marrow chimeras where we lacked PTPN2 in the entire hematopoietic compartment, we saw that that led to clearance. And we implanted animals that lacked PTPN2 in all hematopoietic cells, that these tumors were cleared. We're looking at two different guides. And the reason why we made this system was to look for guides that could benefit anti-PD1. So here we chose a challenging tumor, B16 melanoma, that does not respond to PD1 alone, shown in the black here. And when we gave anti-PD1 to bone marrow chimeras that lacked PTPN2 in the hematopoietic compartment, we found that a quarter of these tumors cleared and the rest were markedly attenuated. And further studies that I don't have time to show you, we found that PTPN2 deleted cells showed increase in their ability to kill, and found that PTPN2 deletion promotes generation and function of cytotoxic TIM3 positive T cells. So to conclude then, we've developed CHIME, a CRISPR-based platform to knock out genes and immune lineages without perturbing immune homeostasis and function. This approach expands the breadth of immune lineages that can be edited, including naive B cells and T cells, macrophages and dendritic cells, and enables in vivo screens in these lineages. We've used CHIME to identify and validate PTPN2 as a regulator of anti-tumor immunity, as I showed you this morning. And our studies established PTPN2 as an attractive cancer immunotherapy target. I showed you that PTPN2 deletion increased cytotoxic CD8 T cell responses and improved immunity to tumors that were poorly responsive to PD1 alone. And we think that PTPN2 deletion can promote anti-tumor immunity by generating and leading to the function of cytotoxic cells. And that in addition, PTPN2 inhibitors can not only improve T cell function, but make tumor cells more vulnerable. And so we think that this approach really enables us to understand new genes and pathways and diverse settings, disease contexts, infection, cancer, autoimmunity, studying a variety of different lineages and therapeutic approaches. And before closing, I'd like to acknowledge the people in my lab this morning. It's been my pleasure and really privileged to have a very talented and committed group of students and fellows. And in particular, I'd like to acknowledge the people presenting work on CHIME this morning. As I mentioned, Marty LaFleur leads this group, and this is the work of Nicole DeRosia, Jasmine DeAndrea, Lauren Milling, Ivy Streeter, Chinsura Kurju, and Sarah Weiss. And finally, it's been my pleasure to have wonderful long-term collaborators, Gordon Freeman, on PD1, John Dench for Juliana's work as Chime, John Weary at UPenn, Vijay Kuchar at the Brigham, Rafi Ahmed at Emory, and I'd like to acknowledge Nick Keening, where a lot of the Chime work started with a wonderful collaboration, and now he is at Arsenal Bio. And finally, thank you for your attention. And I'll stop sharing my screen, and I'm happy to answer questions as time permits.
Arlene, thank you so much for this really wonderful talk. We had quite a few questions from the audience. Feel free to put into more questions. Also for the speakers, if you have questions, feel free to raise your hand. So we can begin with a couple questions from the Q&A box first. So the first one from Philip Yan is about the PD-1 expression on tumor cells. So it's been noticed that recently that PD-1 can be also expressed on cancer cells. What do you think about the role of cancer cell-derived PD-1 and how that regulates T-cell immunity in general?
Yeah, so that's a good question. PD-1 can be expound on cancer cells, on some cells, in particular melanoma cells. And we really don't know as much about how PD-1 is exerting its effects on cancer cells. Is it modulating functions of a different growth factor receptor? In addition, we know that PD-L1, while it's expressed on tumor cells, that it not only is talking to PD-1 on a T cell, but recent work has shown that PD-L1 can signal into tumor cells themselves. It can interact with HRAS, for example, and control signaling in tumor cells and cell motility. So this is a complex network.
Yeah, very fascinating. And a second question from Stan Gu is more about how PTPN2 functions. I wonder if the PTPN2 deletion alters epigenetic landscape in the exhausted T cells, and whether this may complement the effects of PD-1 blockade.
Yes, no, that's a great question. We've done other work to show that some of the way PTPN2 is exerting its effects in T cells is through modulating type 1 interferons. And if we deliver IFNAR, an anti-type 1 inhibitor antibody, that we can overcome this response. So we think it's working in large part through interferons.
Great. Maybe the last question for this session, I think question from Shami Yasushami. I think his question is more general about the future of immunology. I think he's curious about whether the future immunology is to support preventing deaths in the result of different diseases such as cancer and pandemic like COVID-19 with different immunological approach.
So I think this is really a golden time in the field of immunology. I think that PD-1 illustrates the value of basic science. When we started this work, we did not do it to cure cancer. Our work was to really understand fundamental mechanisms of how the immune system is regulated. And I think this is beginning to pay off in cancer, in vaccines, and autoimmunity. And I think you'll hear from all the speakers in this session how understanding fundamental immunology now is being translated. And I think with COVID-19, we're now learning a lot more about the human immune system. It really boggles my mind that so many people, for example, are carrying antibodies to interferons, and how now we can take that understanding and go back to the lab bench and understand what that means in the context of vaccines and cancer. So I think it's really an exciting time in the field.
Yeah, thank you so much, Arlene. For limited time, we couldn't answer all the questions from the chat box. If you have more questions, feel free to reach out to Arlene offline. So our next speaker is Chen Dong. Chen Dong is professor at Shanghai Jiao Tong University School of Medicine, and also the director for the Shanghai Immunotherapy Institute. Chen is also a principal investigator at Institute for Immunology at Tsinghua University. And I think, as many of you know, before returning to China, Chen served as a professor of immunology at University of Texas MD Anderson Cancer Center, and he served as the director for the Center for Inflammation and Cancer. Between 2016 and 2020, Chen served as the dean of Tsinghua University School of Medicine and recruited numerous talented to really build up a wonderful biomedical research at Tsinghua. Chen's taking on an exciting journey now to build Shanghai Immunotherapy Institute. Dr. Dong's research has been to first understand the molecular mechanisms of normal immune and inflammatory responses and apply this knowledge to study pathological response, such as infection, autoimmunity, and allergic disorders. Dr. Dong has made numerous landmark discoveries, including Th17 cells and T follicle helper cells. Many of his works are really fundamental to understand the biological and pathological functions of these T cell subsets. Dr. Dong received numerous awards as well, including the BD Bioscience Investigator Award from the American Association of Immunology, the William Paul Award from International Cytokine and Interferon Society. He's a member of the Chinese Academy of Sciences, a fellow of the American Association for the Advancement of Science and the Chinese Academy of Medicine. Dong, thank you so much for being here with us today and look forward to your talk.
Okay, thank you, Xu. I would like to also thank Harvard CSSA for inviting me to give this talk from a long distance. Since my talk is between Arlene's and David's, I try to make my talk relevant to both on the topic of T follicular helper cells. You already heard from Arlene about co-stimulation. T cells receive a T cell receives many signals from the antigen presenting cells, in addition to TCR recognition of specific peptide presented by MHC molecule, so-called signal 1. You already heard from Arlene that negative and positive co-stimulatory signals that will ultimately determine the T cell fate, activation versus T cell tolerance, exhaustion, and so on. So let me start by telling you about ICOS. ICOS is a CD28 like positive co-stimulatory receptor. Why ICOS? Because in 2001, 3 groups, including Richard Flavel's group, where I was a postdoc to publish this work, along with Arlene Sharpe's group and the TechMax group, showed the knockout phenotypes of ICOS. ICOS is important for T cell activation and T cell function, and especially in terms of T cell mediated germinal center reactions. Germinal center is a structure where B cells proliferate and hypermutate the immunoglobulin genes and differentiate into plasma cells and memory cells. So this reaction, this process, requires CD4 T cells, and the CD4 T cells in germinal centers are also called T follicular helper cells. They were found in mouse and human. First, they were described using the marker CXCR5, which allows T cells to localize in germinal centers and B cell follicles to interact with B cells. And very interesting, after all papers, that has been shown in mouse and human system that TFH development requires ICOS. So that provides some explanation why ICOS knockout were defective in the germinal center reactions. TFH cells had expressed IL-21 cytokine. This is a cytokine that regulates B cell proliferation and germinal center reactions. And we also found that ICOS regulates the production of IL-21 in CD4 T cells. So in 2008, our group by Rosa Nerieva showed that TFH cells express different genes from Th1, Th2, or Th17 cells, and do not depend on these cells to develop. So we hypothesized that this may be a distinct subset of CD4 T cells with importance in germinal center reaction. And in 2009, our group, as well as the labs of Shane Crotty and Caroline Villusa, established BCL6 to be the lineage specific or TFH specific transcription factors for their development. TFH cells not only highly express BCL6, but also BCL6 deficient CD4 cells cannot help B cells to form germinal centers. So therefore, BCL6 is the TFH specific transcription factor. Interesting, in germinal centers, there are two other types of lymphocytes, GCB cells and also T follicular regulatory cells, also highly express BCL6. BCL6 expression in these cells is also required for the development. So therefore, BCL6 really is the master regulator for GC reaction. But how does BCL6 regulate TFH development? Originally, we thought that maybe BCL6 specifically regulates CXCR5 expression, but it turned out from the work of former postdoc Chen Dong, BCL6 does not directly bind to the gene of CXCR5. So we went on to identify two other transcription factors, ACL2 and TOX2, which are highly expressed in TFH cells. By mapping the genes, these three transcription factors directly bind and also regulate. We found out that although these three factors only have two genes to be commonly regulated by three transcription factors, but they also share many between them. So for example, TOX2 and ACL2 together regulates the expression of CXCR5. So therefore, we believe these three transcription factors really form the network to regulate TFH cell development at the transcription level. But how do you get naive T cells to become TFH cells? So I already told you that the signal 1 and the signal 2, but also signal 3 cytokines are also important to regulate helper T cell differentiation. We show that IL-6 can induce the expression of TOX2 and BCL6, and also IL-2 is a negative regulator for TFH cell development by inducing BLIMP1 expression. So therefore, we can induce in vitro TFH cell development by using stimulating antibody to TCR, CD28, and also by using cytokine IL-6 and plus neutralization antibody to IL-2. But this condition was not optimum in vitro. We don't get very high percentages of T cells becoming TFH cells. So therefore, several years ago, former graduate student in the lab, Shiyuan Wang, tried to optimize the condition. So he found that when he used anti-CD3, CD28 to induce together with cytokines, and he found that there's a combination of two chemicals, phosphoester and calcium ionophore. These normally are used to mimic TCR and co-stimulation signals, but Shiyuan find that PI, this combination gives a stronger TFH development than CD3, CD28 combination, either in the neutral condition or in the TFH condition I just described to you by characterizing BCL6 expression and as well as CXCR5 mRNA expression. So Shiyuan then performed RNA-seq analysis and found that comparing T cells activated in the presence of CD3, CD28 plus the cytokine cocktail, PI can stimulate better TFH development at the transcription level. You see genes related to TFH development such as TCF7, IL-6 receptor, BCL6, TOX2, and the CXCR5 were all highly expressed after stimulation by PI instead of CD3, CD28. So then we want to understand why PI was more potent. So Suyuan took inhibitors to TCR pathways, and he found that when he neutralized, when he inhibited PI3 kinase activity, BCL6 expression was upregulated, but CXCL5 expression was downregulated. But on the other hand, inhibitor to MAC, which would activate ERK pathway, can induce increase the expression of both BCL6 and CXCR5. BCL6 protein level was also upregulated. So this suggests that ERK may negatively regulate TFH development. So this was the case because when Suyuan used ERK1 deficient conditional knockout mouse, he found the increase TFH differentiation in vivo. And then when he performed RNA-seq analysis on in vitro differentiated TFH cells, again, we found inhibition of ERK pathway could lead to upregulation of TFH associated genes in vitro. So all of these indicates that ERK negatively regulates TFH development. So then why CD3, CD28 was less potent than PI in inducing TFH development? Suyuan compared the ability of CD28 and ICOS co-stimulation to regulate ERK activation. So you can tell that initially, this is hours at the initial activation after CD3, CD28 or CD3 ICOS co-stimulation. Initial ERK level was similar, but after this, ICOS did not further enhance ERK level, phosphorylation level, but CD28 can further increase ERK activation. And so this suggests that ICOS and CD28 differ in their abilities to activate ERK. So then when we compare T cells activated by CD3 plus CD28 or CD3 plus anti-ICOS, CD3 plus ICOS more potently activate BCL6 protein expression. So therefore, this explains why ICOS was important for TFH development in vivo. So then I told you that ERK deficient T cells, ERK1 deficient T cells were more potent to show increased TFH development, but this was dependent on ICOS because when we cross ICOS knockout onto the ERK1 knockout mice, so they do not show increased differentiation to TFH cells. And since ICOS was required for TFH development, Suyuan put ICOS, Y-type ICOS back into the knockout T cells using retrovirus and transfer these cells in vivo for TFH development. He observed rescue of or even increase of TFH development in the presence of ICOS reconstitution. But on the other hand, when Suyuan utilized a mutant or chimeric ICOS receptor with CD28 cytoplasmic tail, but not ICOS cytoplasmic tail, so the ability of ICOS to induce TFH development was greatly reduced. So this again proves that CD28 and ICOS differentially regulates ERK activity and TFH development. So how does ERK regulates or negatively regulates TFH development? So Suyuan combined three data sets, mature in vivo generated TFH cells. These are all RNA-seq data sets, all pre-TFH cells, because CD3 and the co-stimulation, TCR and co-stimulation would induce pre-TFH commitment. And then PI regulated genes. So there are 23 commonly upregulated genes. So these include BCL6, CXCR5, TCF7, no surprising to you, but there's a new transcription factor we hardly notice called ZAP831. So this was interesting to Suyuan. Of course, he found that ERK knockout show increased expression of this transcription factor, and this is transcription factor localized into the nucleus, and the knockdown of this transcription factor in ERK1 deficient T cells dampen BCL6 and TCF7 expression, suggesting that ERK may negatively regulate TFH development through this factor. So then Suyuan went on to knock down by shRNA ZAP831. He saw that there was downregulation of BCL6, TCF7, TOX2, and so on. And then he combined RNA-seq data of this knockdown with ERK deficient T cells, PI-stimulated T cells, or in vivo generated TFH cells. There are 46 genes shared by all these data sets in the regulation. The upregulated genes include BCL6, TCF7, TOX2, and ZAP831 in itself. And so suggesting that ZAP831 really work together with these pathways to regulate TFH development. But how does it regulate TFH development? So Suyuan made a knock-in flag tag ZAP831 mutant mouse, and use these T cells from these mice to do chip-seq, and then identify the genes that can be directly bound by ZAP831. These include what ZAP831 could upregulate and directly bound 718 genes, including BCL6, TCF7, etc. These are TFH-associated genes. And genes also could be downregulated by ZAP831. There are also 731 genes. And these include genes related to IL-2 signaling, IL-12 signaling, and the TH17 signaling. And then when we compare genes that could be regulated by ZAP831 with those regulated by ASL2, BCL6, there's also a network of shared genes that could be regulated by them. So this suggests that ZAP831 is an important transcription factors that can regulate at the transcriptional level TFH development together with other transcription factors. But how important is ZAP831 during TFH cell development? Suyuan made a knockout in addition to knockdown approach, but he only saw very modest phenotype. And he also noticed there are two other genes that are related to ZAP831 called HIV-EP2, HIV-EP3. ZAP831 is also called HIV-EP1. So then Suyuan decided to knock down these genes and found that knockdown of HIV-EP2 in ZAP831 knockout cells show strongest phenotype. So and also GC response was greatly inhibited. Also antibody production was inhibited. So therefore, we believe that CD28 and ICOS, these two transcription these two co-stimulatory receptors differ in the activation of ERK. ICOS could activate PI3 kinase, but not ERK. And ERK negatively regulates ZAP831 expression. This is a new transcription regulator of TFH development. It can directly upregulate BCL6 expression. And This explains why ICOS is really important as one of the positive co-stimulatory receptor in TFH cell development. And in the last maybe 2 or 3 minutes, let me talk about the role of TFH in human diseases. Particularly, we already appreciate TFH cells are very important in our host defense against infectious diseases, including COVID. Also, GC is a place for a lot of B-cell lymphoma to develop. But of course, Arlene, David, and I have long-standing interest in autoimmune diseases. Related to David's work, TFH cells reported to increase in neuroinflammatory diseases, including one that is called NMOSD. You can read the full name, and these are 2 papers showing increased TFH cells in patients with this disease. And you'll hear from David that these disease, as well as multiple sclerosis, can be treated by B-cell depletion antibodies, one of the major treatment for this for patients. On the other hand, look at minimum for MS is the most commonly used is to 55 peptide AE disease. We know that this is independent cells, and also postdoc in the lab, Lin Zhang, also examined TFH, and therefore, unlike human disease, this animal model we know is largely dependent on Th17 cells, but not B cells or TFH cells. So we turn to a different model, has been reported with a whole protein, recombinant protein, to induce EAE. So we compare these 2 models and found that that induced by the whole protein also induced much stronger GC reaction as well as TFH development. And this model induced by the whole protein was reported to depend on B cells. So therefore, Lin Zhang tested whether or not TFH cells were required in this model. And she found that in the absence of TFH cells, when she utilized BCL6 conditional knockout mice, never got any disease. And also, as you can imagine, TFH cell in draining lymph node and CNS were all greatly reduced or gone. So therefore, present the pathogens and the MOSD disease, and we are currently investigating the function of TFH cells as well as cells in this model. So lastly, let me conclude by acknowledging the lab members and the collaborators. One or the student who did the PI, the ZAP831 story, and Lin Zhang is currently working on the whole mark protein-induced EAE disease. Xiaohong Zhao is a bioinformatic analyst or lab. She helped us in his bioinformatic analysis, and Xiaohu Wang me to these projects. We got help from the mark for years. So let me add, and also maybe I can.
Thank you, Chen, for the really amazing talk. Please feel free to questions or raise your hands for the panelists. I can begin with a couple of questions. It's really interesting to see the ZFP proteins relating the T cell subsite program. Is a
So other primary regular errors, gene. So we know that unregulated genes, so we still don't know the mechanism. Yeah,
it's very interesting.
we know.
Very interesting. Yeah. Another kind of really general question is, what kind of task can be generally induced as well? Do you think in other stress conditions which also impact T cell polarization, would they also use or leverage similar mechanisms in that process?
Yeah, so that's a very good question. I'm a relatively stress-free person, so I don't care about stress that much. But we TFH cell plasticity in different infectious disease models as well as autoimmune disease models, and it's very obvious that in infection, depending on the, for example, type 1 intracellular bacteria or viruses, you can start to, in these models, you observe interferon gamma or Th1-like TFH said, due to the bona fide TFH or Th1 cells. And these these T cells with dual programs, we reported recently necessary for making neutralizing antibodies in infections. And also and others observed in response to helminth infection or allergens, TFH cells could also upregulate Th2 associated gene program in them, and which may be also important for production of high affinity IgE.
And maybe for the consideration of time, one last question for Chen is, do you think targeting BCL6 may be able to specifically target?
So that's a good question. BCL6 is also expressed by TFR cells. So if you target BCL6, of course, you can wipe out or you can inhibit TFH program, but you may also interfere with TFR cells. We start to observe BCL6 expression in many T cells as well as lymphocyte populations. So, so before that, I would be cautious about targeting BCL6. And also the difficulty is how you target a transcription factor. Maybe PROTAC, that kind of method will be feasible, will be possible, but I will still be cautious about test before we know everything about BCL6.
Yeah, thank you, Chen, for joining us remotely, very late on the Saturday evening back there in China. So now we're moving to our last speaker of this really exciting morning session, Dr. David Haffler. David is trained as a physician scientist. He's currently the William and Louis Stiles Adjunct Professor and Chairman of the Department of Neurology, and also professors of immunobiology at Yale School of Medicine. He's also the neurologist in chief of the Yale New Haven Hospital. Dr. Hafler has made many seminal discoveries defining the pathogenesis of multiple sclerosis and autoimmunity. He co-led the discovery of genetic variants causing MS and demonstrated how these variants alters immune response in relation to the environmental factors. The work shows that how to solve this is really inspiration for the field to environmental factors. His work many areas, including understanding new viral-induced diseases, also to cancer immunology. Dr. Hafler received a Jacob Javits Merit Award from NIH, Distal Prize from Sclerosis Research from American Neurology, the Ryan Adam Prize from the American Neurologists Association, and was the 2016 Frontier Lecture for the American. Has been elected to the National Academy of Clinical Investigation, the Association of American Physicians. Welcome here, Dr. Hafler. Thank you so much for joining. I look forward to your talk.
Thank you very much. Pleasure to be here today. Great to see you. It's been a while. Hopefully we can all start traveling again, and this horrid pandemic will be coming. It's Father's Day this weekend, and I'm on the island man with my granddaughter, and I apologize up front. Hopefully that not an issue this morning. Anyway, so the title of my talk is Autoimmune Disease, a 1,000-foot view of autoimmune using MS as a model autoimmune disease. The autoimmune diseases are in fact complex genetic diseases where the genetic variants map almost exclusively to the immune system. In MS, it's believed that B cells, discussing, drive the inflamed myelolytic system. I'll show that T cell traffic with blood and do inflammatory gamma interferon type signature, which we believe is involved in homeostasis in the central nervous system. Then show again data which revealed novel immune pathways that we believe drive multiple sclerosis, the foundation for generation sign of more precise immunogenesis might be for disease. Show that the signature is also found in healthy brain. So first, the background of the genetics. And question is, what are the genes that cause disease? And basically, there are two types of genetic diseases. One where you have gene knockouts. Example would be muscular dystrophy. Dystrophin gene is knocked out, the muscles can't form, and it's a very highly penetrant mutation. But it's common diseases, be it the autoimmune diseases, hypertension, cancer, many small effects become a large effect. So you have multiple genotypes, again, each with a very small effect of variants. Okay, allelic variant, which have a very small effect. These people, an example shows published a variant, the haplotype in NF-kappa. If you have that variant, with about 20% of the population, more frequent in MS, you have a 1.2. If you are carrier of that variant, the risk variant, you have a 20-fold increase in NF-kappa BX. So small, but the phenotypes are really quite remarkable. These phenotypes interact with the environment. It's not bad genes, not a bad environment, but the bad interaction with genetics, which involve microbiome. We know diet, as was mentioned in the introduction, salt, and these lead to autoimmunity. And here I'll discuss multiple sclerosis. Here back in 2007, we published among the first first GWAS scans performed, done at the Broad Institute, and led by myself, Steve Hauser, and Eric Lander. And we were to identify 3 genetic variants significance in different conditions they're being associated with disease risk. We've since gone on now and have looked at over 47,000 patients with MS, 6,000 to 8,000 control subjects, all very carefully matched genetically. whereas 5 years ago, we've now identified about half of the MS heritability with 233 genetic variants. These are common variants. And we identify variants associated with MS risk. So one can look at a chart like that and say, well, what do you do with those data? That is, how did the genetic variants cause the disease? So how do you go from tagging SNPs? And again, the actual SNP doesn't mean it's tagging. Go from the pathway mechanisms. Let me show an example of work by Alex Marston and Institute. So what one can do is to make a map of the chromatid that's open. So here's a basic experiment. If everywhere you have open chromatid, you have a SNP or a haplotype associated with disease risk in a particular cell type, that cell type is likely to be important for the disease. On the other hand, if every place there's a haplotype, or let's say a SNP, which is in the genetic region is closed, the chromatid is inaccessible, is measured by K27 acetylation, then that cell type is unlikely to be important. So here in this slide, you can see different cell types, including brain, different immune cells. Part of the ENCODE project, our lab generated the K27 acetylation, K4 methylation maps, the different T cell populations, other tissues, including kidney, osteoblast, liver, and so you have different diseases going from Alzheimer's disease to renal dysfunction and high BUN levels. So one can see in the upper left corner that in the various neurologic diseases, you have genetic variants hitting parts of the brain. Here, concentrating on multiple sclerosis, you see that the genetic variants associated with risk of developing MS are hitting open chromatid in T cells, predominantly Tregs, Th17 cells, CD8 cells, but also B cells. In fact, there are only 3 diseases where the B cells were hit, include primary biliary cirrhosis and systemic lupus. Other autoimmune diseases do not have genetic variants, by and large, hitting the B cells. And here we have these hits here in the B cells. Now, why is this of interest? Well, as Chen just mentioned, MS therapy has been revolutionized by the use of B-cell depleting agents. This is the clinical trial looking at relapsing remitting MS with anti-B-cell monoclonal antibodies. Work was led by my former colleague from Harvard when we were postdocs together, Steve Hafler, using the drug Ocrevus, which is basically Rituxan, with a modification which is humanized rather than with a chimeric antibody. And one can see this dramatic effect of a 98, 97% decrease compared to the other previous therapy, beta interferon, mean number of patients with positive MRI scans. I must say we have virtually all of our new onset patients on ocrelizumab, and it's very rare for a patient to break through. So in fact, the genetics predicted that B cells would be important in MS, but of course the clinical trial was done around the time we did the B cell work. But it's nice that all of it comes together. So do we know the cause of autoimmune disease, i.e. multiple sclerosis? You never know causality. You don't know what you don't know, but I would argue based on the genetics, based on the animal models, based on the ex vivo and work in terms of T cell reactivity to self-antigens, and most importantly, based on the clinical trials showing a clear effect of immunosuppression on the disease, we have a good working model for autoimmune disease and MS, which of course will continue to have more and more refinement. So what's the working model for autoimmune disease? In MS, we believe it's a genetically mediated autoimmune disease initiated in the periphery by T cells, B cells, and macrophages that traffic is. If you block T cell traffic with anti-BLA4, you also you stop disease progression. So the thought is that antigens cross-react with myelin, perhaps EBV is recently presented by the Bill Robinson paper Nature, with co-stimulatory molecules being the adjuvant IL-12, IL-23, leading to activation of myelin-reactive T cells. We all have myelin-reactive T cells in our circulation. We can find T cells reactive to MOG, myelin basic protein, protolipoprotein, and virtually every healthy individual. And these are high affinity T cells. It's believed that the regulatory T cells, FOXP3 positive Tregs, prevent the activation of these autoreactive T cells in health. And here is a paper published by Visi Vigletta in the JEM back in 2004, which was the first demonstration that there are defects in suppression by regulatory T cells in patients with an autoimmune disease, shown defects in rheumatoid arthritis autoimmune diseases. So here we have patients with MS and healthy donors in the closed square. These are all age-matched, these are all untreated patients. This has been looked at as decrease in the ability of in vitro assays for T cells to be blocked, to Tregs to have loss of function. And what is the mechanism for this loss of function? Here we have Tregs from untreated MS patients secrete gamma interferon. Again, this has been shown subsequently in other autoimmune diseases. We have an example of a control of an MS patient where we isolated FOXP3 positive, ISCD25 high, 127 low, Tregs for ex vivo, stimulated for 4 hours with ionomycin PMA, and one can see these increases in gamma interferon, from serious and controls. This high gamma interferon secretion in Treg, a decrease in Treg in T cell activation, autoreactive T cells also say that the TR1 cells secreting IL-10. But it was a bit surprising to see these myelin-reactive T cells in healthy individuals, and just to show that they do exist, but rather than secreting inflammatory cytokines such as IL-17 and gamma interferon and GM-CSF, they secrete IL-10, and I'll show work. This is from Will Cow, published in Science Translational Medicine some 7 years ago now. We did single clone analysis of myelin-reactive CCR6 positive T cells, MS patient controls, and you can see that in MS patients are secreting these inflammatory control subjects secreting predominantly IL-10. So we have, and then the interesting question arises is why do we have autoreactive T cells secreting IL-10, and we speculate they may be involved in scar formation, for example, when has a stroke. These autoreactive T cells may help in scar formation. To summarize, B cells appear to be the antigen-presenting cells that drives inflammatory, myelin-specific CD4 T cells into the central nervous system, causing the disease. And also mentioned in a paper recently submitted, which is recently presented, in terms of the molecular mechanism, next layer mechanism for the increased gamma interferon secreting Tregs, is that a short form of PRDM1 blocks a long form of PRDM1, which is induced both by genetic and environmental factors, in particular salt, which induces SGK1, leading to dysfunction of these Tregs. So what about T cells going into the brain? The T cell traffic between blood to tissue is very tightly regulated. So a very important point that's evolving over the past few years is that tissues develop a unique homeostatic immune state. So for example, in the skin, gut, or adipose tissue, the very specific factors and chemokine receptors which mediate traffic into the tissue. What about between blood and CSF? Well, we find there are T cells with a unique blood T cell state, then an overlapping cell state as they migrate through into the central nervous system through the choroid plexus, and then a unique CSF T cell state. I'll show some of these data now. But the important point is that this cell with identical T cell receptors, as it traffics into the central nervous system, it is driven by the milieu in the central nervous system to acquire unique features. So I want to acknowledge Jenna Papalardo, an incredibly talented graduate student, and this is her PhD thesis. We performed the first transcriptomic and clonal characterization of T cells in the human central nervous system. Basically, we took spinal fluid from patients with MS, isolated the blood and pelted the CSF, and did 5' Illuminex single-cell RNA sequencing, and and then analyzed the subjects. We did the first 6 healthy donors and 5 patients with MS, all age matched. And to say that we've now done over 30 MS patients and 12 healthy controls, and those data now being analyzed in the context of a clinical trial with anti-CD20 and B-cell depletion. Looked at over 100,000 cells and over 50,000 T cell receptors. First, there was, as previously shown, I'll show in a moment, the predominant cells in the spinal fluid this is blood and this is spinal fluid. The predominant cells in the spinal fluid are T cells with small numbers, dendritic cells, and some very unique CSF-specific monocytes that look a bit like microglia. In fact, when Jenna went back, she goes, gee, you showed this 25, 30 years ago. We first showed that there are inactivated T cells in the blood and spinal fluid of patients with MS. So we started to examine the spinal fluid from patients with MS, and it was very inflamed. It's not surprising. This is Jenna Papalardo, and it said, we need to do controls. So went back and we did spinal taps on 12 healthy Yale grad students and postdocs. And just to be clear, I have no idea who they were. There's no coercion or collusion in performing these spinal taps. So in healthy spinal fluid, the T cell surprisingly acquired an inflammatory gamma interferon type signature. So here's a global, and so we examined the global structure, which revealed a blood to CSF continuum of T cells. As I said, when T cells traffic from the circulation into the nervous system, they acquire certain characteristics as they enter into the nervous system. So here in red, we have blood, and in blue, we have spinal fluid. And we used a tool developed by Smita Krishnavami, where the cell progression of blood and CSF is revealed by FATE. So FATE is a potential of heat diffusion based setting. It's an unsupervised low visualization method that captures nonlinear structure using an information geometric distance between data points. So here we have the original tissue, blood, and spinal fluid. So here we have the CD4 cells here and CD8 cells here. Using FATE, you can see the high expression here of CD8 cells and CD4 cells as they transition into the CSF. So we first use a program called Phenograph, where we divide the different populations looking at the top 10 differentially expressed genes in each cluster. And we then visualize these healthy T cells colored by this phenograph here, looking at the cluster between blood and CSF. So here, for example, we have very naive CD4 cells. Here are cells which are in transition, and here are cells which are very much embedded in the nervous system. And here the same for CD8 cells. And again, all this has been published. I'm just giving the overview of these data. We then, the original identity of the cells to generate a continuous is a probability transitioning from one cell to another in a random walk. So it's a readout of cell-to-cell similarity. So here cells are very blood-like, here are cells which are very brain-like, and you can see the transition of CD4 and CD8 cells as they become more CSF-like. So again, the tissue score is how CSF-like or how blood-like a cell is. And then we ask what distinguishes these different states using this tissue. So I'll give an example. Here are 3 different markers. ITGA4 that is alpha-4 integrin, or as I mentioned earlier, VLA, part of VLA4, which is required for T cell traffic into the nervous system. It's a target of natalizumab, which treats MS. And one can see that, again, high expression is in orange and yellow, that the alpha-4 integrin is highly expressed in the CSF. CXCR3 is also highly expressed on T cells in the spinal fluid and in the brain. And you can see, again, very little expression in the blood. But CCR7 is a chemokine expressed naive T cells, which excluded from the CNS. So you can then look at T cells in healthy spinal fluid. In many respects, I found the results looking at healthy spinal fluid as interesting as looking at patients with MS. So one that characterizes blood CSF continuum, this is a heat map of shape-based gene clusters. Again, I won't go into details of how we did this, but basically the gene expression was imputed with a tool called MAGIC, also from SMITA, to visualize gene expression patterns across different tissue score. There is so much data to mine here, but let me just show one example what we could do with the 9 clusters based on the tissue score. And in particular, we're intrigued by this Th1 cluster here in CSF-like cells with expression of gamma interferon, Tbet, granzyme K, and almost other factors involved in gamma interferon secretion. So why should there be gamma interferon secreting cells? So here are some of the genes related to the CD4 and CD8 clusters. Again, looking at Th1, particularly in the CSF memory cluster 3. These are heat maps showing the Z-score for each gene, the gene module, with very high amounts of these gamma interferon, CXCR3, Tbet, IL-12 receptor, interferon gamma. Then we have another population with measures of tissue residence, including BLIMP1, PRDM1, CD69, LAG3, CXCR6, all measures of tissue residence. And then measures of cytotoxicity, including granzyme K, I mentioned it, and other markers of cytotoxicity. Again, these are all CD4 cells from healthy spinal fluid. CD8, again, we have measures of tissue residence and measures of cytotoxicity. So just to summarize all these data, what are the predicted patterns of T cell composition, blood, and CSF? So the markers where over here, where cells are excluded from entering the spinal fluid. Then over here, cells which enrich for traits necessary for entry into the CSF. And finally, have not yet entered and gone specific changes, then CSF entry and tissue-dependent changes. These are cells that have been very much conditioned in the central nervous system. And if we look at the major genes we found, Th1 genes, found resident markers of resident T cells, ones involved in cholesterol homeostasis. Not surprising considering 80% of the brain is fat tissue and cholesterol. Genes involved in TGF-beta pathway, and then co-inhibitory receptors. And I'll discuss this in just a moment. So taking those various data from single cell analysis, it becomes critical to translate it from hypotheses into experiments. I think once you view the single cell data as hypothesis generating, that needs to be validated at the protein level. Again, there are all these data in the public domain for everyone to analyze and much to potentially learn. So in terms of gamma interferon, so here's looking at spinal fluid and blood. This is PD1 gamma interferon after 4 hours of stimulation. You can see that in blood, there is only about 4% of the cells secreting gamma interferon, only about 2% that PD1 gamma positive. But in the spinal fluid, almost half the cells are producing gamma interferon. Really quite remarkable. Here's a summary of 3 different subjects. I think it was curious of what is this phenotype of PD1 high gamma interferon. The work we published a number of years earlier by Brittany Goods, looking at PD1 high effector cells. So these are looking at total CD4 cells, PD1 negative, and PD1 positive, that engage very high levels of PD1. And you can see if you stimulate these cells, they do not enter cell cycle, yet they secrete marked amounts of gamma interferon. And in fact, it leads us to speculate that immune privilege in the central nervous system may relate to the high expression of co-inhibitory molecules which will secrete gamma interferon, perhaps involved in homeostasis in the brain, yet they do not enter the cell cycle. So now that we know the normal spinal fluid has an inflammatory signature, what about autoimmune disease with MS? So using the single-cell RNA-seq analysis, it revealed novel immune pathways that drive MS. So first, there are no significant differences between conventional T-cell types and healthy controls in MS. Here are the different populations I discussed earlier in MS or control CSF of the CD4 and CD8 populations. So one can see they're very similar, no marked differences. Again, this is all in the paper, but the CSF3 are the ones that are predominantly making the gamma interferon in MS and controls. This CSF2 population is one that does not appear to have much in the way of tissue-resident markers, and we believe goes in and out of the spinal fluid of the nervous system. So we looked at the top differentially expressed genes in T-cell clusters between MS patients and controls. And what we found is that there are a number of genes that are associated with MS. Again, these are the T-cell clusters with log-fold changes in blood and CSF. I'll just point out some of them of interest. MALAT1 is an epigenetic modulator of gene expression and regulates cell motility, is markedly increased in MS. IL-32 is a pro-inflammatory cytokine that induces TNF-alpha and IL-6. In fact, one paper previously published showing increased IL-32, but something we had not previously looked at before, which is also markedly increased in the T cells and MS spinal fluid. I just added granzyme K, and I just highlighted this after a wonderful talk by Michael Brenner, a colleague of mine from the Brigham, who just recently showed that granzyme K is associated with complement cleavage and involving complement activation in CD4 and CD8 cells. So this would suggest that the granzyme K that we're seeing highly expressed in MS T cells may in fact be involved to some degree in complement activation in the nervous system. And June B, part of the AP1 transcription factor involved in T cell activation. And as I mentioned earlier about the short form of PRDM1, the PRDM1 short form induces FOS-June, that is AP1, as part of the mechanism for loss of Treg function and in patients with MS. Well, I showed you spinal fluid. What about the brain in healthy individuals? So there, in fact, is an inflammatory Th1 signature also seen in healthy brain. So here we characterize T cells in human brain parenchyma. Again, using the phenograph, we identify different populations, including a small population of T cells. Looking at the lineage tissue residence functional gene expression, T cells, again, within normal brain parenchyma, single cell RNA-seq, we found a very strong overlap between what we found in brain T cells and CSF T cells, again, in healthy subjects. What's interesting is, again, we see this RNA signature in T cells. This is from RNA from single cell analysis of brain, and this is doing Yingzi from healthy brain. But we can see this significant gamma interferon signature and this granzyme signature in normal CD4 cells. We're also with Andrew Wang, a very talented graduate student, Tomimi Yoshida. We're looking at T cells isolated from mouse brain. Our lab is basically a human lab, but there are important times to use mouse models when there's specific questions to ask, when you can't ask the question humans. And just to show that we can isolating T cells from normal mouse brain, you can see this gamma interferon signature, both CD4 and CD8 cells. These are just normal mice. And here's the gamma interferon, which is seen in the brain, but not in other tissues. And it won't show these data, but basically what what Tomimi found was that if you put these animals in the germ-free condition, this gamma signature basically disappears, indicating that there is a gut-brain axis, as Vijay Kutru recently showed, which is likely driven by the gut microbiota. So as a selective migration, Th1 cells are shaped in the CNS, that is their glial T-cell communication circuit, is known from the late Ben Barrett's astrocyte condition microglia. The astrocyte-derived TGF-beta and cholesterol required for microglial survival, and that soluble factors produced by astrocytes, including cholesterol lipoproteins, which are found in the CSF. So we took astrocyte conditioned media where normal CD3 cells can show an increase in gamma interferon, and with cholesterol and TGF-beta could show an increase in gamma, again from healthy CD3, CD4 positive, CXCR3 positive cells. So again, we believe this cabinet signature we're seeing in normal brain is being mediated by astrocytes through cholesterol and TGF-beta. What might this mean for the role of gamma interferon in brain homeostasis? The known functions of gamma interferon in the central nervous system, including chemokine production, neuroprotective functions such as glutamate clearance, neuronal survival, and we speculate synaptic pruning by influencing microglia. In fact, it was shown by a number of years ago by Jan and Kipnis that if you knock out the gamma interferon, that animals develop a depressive type syndrome with changes in social behavior. So I think I wanted to sort of point out how single cell analysis can take us down different areas of investigation, suggesting that how these pathways involved in homeostasis, that is, the immune system is much more than just fighting off microbes, but is critically involved in homeostasis of different systems, and here in the central nervous system. So in summary, autoimmune diseases are complex genetic diseases where genetic variants map to the immune system, and MS B cells drive inflamed monospecific CD4 cells in the CNS. This traffic between blood and spinal fluid is tightly regulated. In healthy CSF, T cells acquire Th1 signature, and the T cells in blood and CSF are functioning different, indicating the CNS shapes the homeostatic T cell state. And there are a significant number of phenotypic differences in health and MS in the more expanded cells in the central nervous system. And this will be an area of, I think, very active investigation, deciphering and generating hypotheses from these data over the next decade. And finally, we saw this Th1 signature in healthy brain. So let me just acknowledge individuals in the lab, in particular Jenna Papalardo, since going to graduate, Li Zhang, who runs a single cell platform in a laboratory, individuals at our Interventional MS Center, Smita Krishnawamy and David Van Dyke from Genetics, Jack Antel and Kelly Perman provide data from a fresh brain, and all the healthy Yale students and who have gone in who have had spinal taps for this project. And here's our group. This is before when we get together. We're finally going to get together again. Please come visit us in New Haven. This is the view from the back of our house to the base of Long Island Sound. Here is our last MS retreat, which we'll have another one in a few weeks. So again, please do visit us, and thank you for allowing me to give this talk this morning.
David, thank you so much for the talk and joining us during your vacation on Father's Day. So we can field a couple of questions. There's a lot of questions in the chat boxes. We can begin with a question from Chen Dong. David, do you see Treg cells in the CSF from healthy patients? I guess a piggyback question is to also see the autoreactive T cells in the healthy patients. Yes.
Oh, it's a fantastic question, and the short answer, we don't know, because the number of cells we can get from the spinal fluid is very low. We've looked at the single cell data with CiteSeq, and the numbers are so small, we're not confident of the data we're getting from the CSF. So it's a really active area of investigation. I suspect the Tregs in the spinal fluid will be making gamma interferon, because normal CD4 cells are making gamma, and it's something we're beginning to look at. So I'm the bet a nickel that they're making gamma, but we haven't addressed that yet because of small numbers of cells.
Another question is, could you comment on the potential role of the meningeal B cells on the CNS T cells?
Oh, it's a fantastic question. So the present thinking is that when you have autoimmune disease, it tends to begin. I'll be taking the beautiful work from Diane Mathis and Christophe Demois and look at the NOD model of diabetes, which is really a good model of diabetes because it reflects the genetics of the disease. Before you have parenchymal involvement, invasion of the islets, you first have the periencephalitis. I believe a similar thing occurs in rheumatoid arthritis, where you have a periarticulitis. We believe the same thing is occurring in MS, where you first have a somewhat of a perimeningitis, which are then secondary involved in parenchymal invasion. We're starting a clinical trial at Yale, which in Nantucket, we're taking patients before they have clinical disease, but have radiologic disease, screening individuals with high genetic risk scores and doing MRIs. It's called radiologically isolated syndrome. But I think that's too late. I think what we really want to do is to go in before there's parenchymal damage. So we're labeling this immunologically isolated syndrome. I don't know how to identify it. I don't know if it exists, but we really like to start doing spinal taps on individuals at risk for MS. To see if we can begin to identify a perimeningitis as the meningeal involvement with B cells as a first step. And another question relates to if there are B cell follicles there, which we believe they are the TFH cells or TPH cells that are in the perimeningeal region, which are driving the.
Thank you, David. For the time's consideration, we couldn't go to all the questions that people put in the chat box. Feel free to reach out to David for follow-up discussions. Thank you, David. Thank you, Chen. Thank you, Arlene, to join us this morning for a fantastic session. So hope you enjoy the rest of the day. We have an amazing lineup of speakers for the rest of the day. Now getting back to the host for the next agenda.
In the next 15 minutes, I will provide an overview and update of our multi-omic solutions in the US market. BGI is a global pioneer in omics research. It was founded in 1999 as a nonprofit institute by participating in the Human Genome Project. Throughout its history, it has contributed to many key milestones in genomics research, including the Rice Genome Project, the International HMS Project, the first Asian genome, and the 1000 Genome Project. In recent years, with spatial genomics, single-cell omics becoming more popular, BGI once again applied its unique technologies on a number of high-impact studies, including the first whole-body cell atlas of non-human primate and the first panoramic atlas of mouse embryo development, published recently as a cover paper on Cell. BGI's vision is omics for all. It aims to develop and apply genomics technologies for the betterment of mankind. BGI was founded in 1999. As of this year, it has over 10,000 employees worldwide. Its headquarters is in Shenzhen, China, with its services and solutions in over 100 countries and regions. The BGI Group is a collection of BGI's nonprofit organizations and for-profit companies. BGI Research is a research institute focusing on genetic research and technology innovation. The GigaScience is an open access publishing division which publishes journal articles, datasets, and software. The BGI College develops educational and training programs. BGI Genomics is BGI's first public company providing genomic research services, diagnostic testing, and IVD solutions in the areas of reproductive health, precision oncology, and pathogen detection. While MGI develops and manufactures genetic sequencers and laboratory automation products, there are also a number of other entities that are in incubation phase right now. BGI Americas is the US subsidiary of BGI Genomics, currently engaged in genomic services, mass spec analysis services to the research service market, and supplying IVD products to the diagnostic markets as well. BGI was founded as a research organization with over 20 years of investment in R&D. As of last year, BGI has more than 3,700 patents worldwide. Nature Index publishes its annual ranking of high-quality research and collaborations. In 2021, BGI was ranked #1 among life science corporations in Asia-Pacific area, and also #10 among global life science organizations. BGI holds high standard for corporate social responsibility. Quoted BGI's chairman Wang Jian, "When a health emergency strikes, BGI has a duty to be the first in line to help." BGI became known to the public in 2003 when it first sequenced and developed the diagnostic kits for SARS virus. In global disaster relief efforts, for example, in the 2004 Indian Ocean tsunami, 2008 Wenchuan earthquake in China, the German E. coli outbreak in 2001, and also 2014, the Ebola virus outbreak in Africa, BGI was always at the forefront. It is not surprising that BGI again became the first responder in 2020 for the COVID pandemic. With its high throughput diagnostic products and laboratory solutions, contributing heavily to help contain the virus on a global scale. Our team in the US was also part of the response team by leading the FDA emergency use authorization of our diagnostic products in the very early phase. So as of today, BGI Americas already has a 12 years history. It was first established in Boston in 2010. In 2011, we built a joint genome center with Children's Hospital of Philadelphia on rare disease research and diagnosis. In the same year, we established a major collaboration with Merck on biomarker and genomic technology development. In 2013, BGI acquired Complete Genomics, which now became the R&D division of MGI. In 2017, our team first introduced a $600 whole genome sequencing in the US market based on our DMBC platform. And in 2018, we built a mass spec analysis service center in San Jose, California. In 2020, we received our first FDA authorization of our COVID IVD product. Evolving from the 2013 acquisition of Complete Genomics, MGI already has a line of sequencing platforms covering a very broad spectrum of sequencing throughput. The most popular sequencers are the DMB-Seq G400 and the DMB-Seq T7. The G400 has a data output of more than 400 gigabytes per run, and the T7 sequencing throughput is 6 terabytes of data based on P150 reads. Our team has been offering DMB-Seq-based sequencing services for almost 5 years since 2017. You may also hear that the news, the MGI plans to start commercializing its sequencing platforms in the US market beginning this August. DMB-Seq is a collection of proprietary technologies underneath our sequencing platforms, including the rolling circle amplification of single-stranded circular DNA, the DNA nanoballs, the patterned array, and the combinatorial probe anchor synthesis called the CPAS. So these technologies deliver unique advantages, including low PCR bias, reduced duplication rates, low index hopping rate, high accuracy compared to the other major sequencing platforms on the market. The compatibility and advantage of our sequencing technologies has already been demonstrated in over 2,000 scientific publications in a wide range of sequencing applications. Our US commercial team has been very actively supporting research customers' adoption of sequencing applications based on DMB-Seq platforms. So far, we can tell that the adoption and the customer feedback has been quite positive. Genomics researchers in the US are generally very convinced of the data quality, compatibility, and quality of service offered by our team. About 4 years ago, we have invested in a state-of-the-art mass spec center in our San Jose facility to better serve our customers' needs for proteomics and multiomics research. Based on the advanced mass spec platform and an experienced team of industry veterans, the laboratory offers a diverse menu of LC-MS analysis services for proteomics, metabolomics, as well as biologics characterization services for our academic and biopharma customers. So by integrating product menu from both NGS and mass spec analysis services, BGI now offers a truly multi-omic solutions for genomics, transcriptomics, epigenomics, proteomics, and metabolomics. We offer a one-stop shop for the most comprehensive research services, including whole genome sequencing, exome sequencing, RNA-seq, single-cell sequencing, whole genome bisulfite-seq, quantitative proteomics, and untargeted metabolomics. Our R&D team has been continuously developing new analysis tools to support the rapidly evolving omics research field. So currently, we already have a list of multi-omic solutions from sample to integrated data analysis. They include on this menu, integrated analysis for proteogenomics, integrated analysis for transcriptomics, proteomics, and metabolomics, and integrated analysis for metagenomics and metabolomics. We hope these combinations provide unique insights to uncover the correlation between these different types of omics datasets. Here are some recent highlights from our local laboratory in San Jose. First, we are able to offer local premium services for RNA-seq and pre-made library sequencing. The lab is now ready to accept pilot projects. Second, we have expanded our local mass spec analysis service menu and now are able to offer fully localized metabolomic services in addition to proteomics. Third, we are actively establishing our local data analysis and data management infrastructure built on AWS. And we will continue to improve our local service workflow to provide convenience and shorter TAT for our customers. And here are a few services to highlight. We offer whole genome sequencing with PCR-free, low-pass, long fragment read protocols, which is a comprehensive portfolio. As I mentioned earlier, that we are about to launch our premium RNA sequencing and pre-made library sequencing services directly from our local laboratory with rapid turnaround time and convenient sample preparation. We also offer end-to-end single-cell RNA sequencing analysis services based on 10x Genomics platform. The Mass Spec Center offers its unique isobaric label TMT and the label-free DIA methods for quantitative proteomics, and also untargeted metabolomics as well, which has unique, large in-house database for metabolites. You may also be aware of our recent breakthrough in spatial genomics, which is a unique technology developed by BGS research team called StereoSeq. Its full name is Spatial Enhanced Resolution Omics Sequencing. StereoSeq provides nanoscale resolution and centimeter-level panoramic field of view for spatial transcriptomic profiling. A series of high-impact papers have already been published in the past months, including the Cell cover paper on spatial temporal transcriptomic atlas of the mouse organogenesis. And I'm glad to inform that the StereoSeq collaboration is now available in our San Jose laboratory, so feel free to contact our team for more details. And lastly, our team has been actively expanding in the US. We now have a series of positions open for sales, marketing, product management, laboratory scientists for both NGS and mass spec services. So feel free to reach out through our recruiting channels to learn more details about these exciting opportunities. We welcome new talents to join us to revolutionize omics together. And thanks again for the opportunity to update BGI's current capabilities. Please feel free to reach out to BGI through our public channel or our local sales manager, Daniel Gobel, based in Boston. Thank you very much, and have a successful event.
He has published more than 180 papers with more than 76,000 citations, which makes him one of the most influential scientists in the world. So it's really our pleasure to have Dr. Zhang here, and let's welcome Zhang, and we look forward to your talk.
All right, great. Thank you very much, Nan, for your nice introduction. So I also want to thank the CSSA, the meeting organizer, for putting me in this session so that I can talk about some of our new work. So, between two highly accomplished neurobiologists, Qiufu and Qiufu, I feel it's an order, and at the same time, it's also make me kind of nervous. Compared with them, I'm kind of an amateur. Okay. Anyway, I'm going to try. So now I share my screen. All right. So you guys can see my screen, right?
Yep.
So basically, today my main talk will be focused on the neurohistogeneity and some aspect of characterization, some sub-neuron type in addictions. So as Nan mentioned, that my group has spent more than a decade to study epigenetics, and particularly in identify and characterize different class of epigenetic enzymes, including two HDAC complex and several histone methyltransfers, and the Jumanji protein family of histone D methylase, and ubiquitin E3 ligase or H2A, as well as the TAT family of diogenes that is involved in removing DNA methylation. So basically, after we demonstrated that TAT family-mediated process can remove DNA methylation, so basically the majority of the epigenetic modifications that are on DNA or histones, the enzyme involved in the adding or removing has been characterized. So we basically have to change to ask other questions. So I decided about 10 years ago to focus on some epigenetic mediated process. So basically, we choose two different directions. One is the epigenetic chromatin remodeling, chromatin reprogramming is the beginning of mammalian life. So basically, ask how the egg factors can remodel a sperm and leading to the early developmental chromatin change. And then similar to that, we also ask how egg factors reprogramming somatic cells, that is, somatic nuclear transfer reprogramming. So during the past 10 years or so, we have made a several important discovery, including identify some of the important factors for the genome activation and identify the barriers that prevent the success of somatic nuclear transfer and found ways to overcome that, as well as discovered a new genomic imprinting mechanism and characterize the function of this new mechanism in development as well as in somatic nuclear transfers. So the other biological process we are interested in is like how epigenetic chromatin modification play a role in the reward learning memory process, which we basically use drug addiction as a model. So why we went so far, the initial interest on this question is triggered by my curiosity as to how this learning memory are formed and what mediated that process. Actually, many years ago, I asked Chen this question. I said, exactly how the memory is mediated, what actually is the molecular base or the materials, physical base mediated that process. So he told me, oh, it's a non-term potentiation, a short-term non-potentiation, which is, and then I asked what mediated that. He said neuroplasticity. So bottom line is that he couldn't give me an answer what's the materials that mediate this process. So I'm wondering whether the epigenetic memory that's, people in our field study have anything to do with real brain learning memory. So that's the initial question. It's quite naive that until after, we started to get into this, I realized actually it's too early to address that question because people even don't know what neuron type, what cell mediates this learning memory process. So that's where we start with. So the approach we basically take focus on the brain reward circuit. So because we are trying to understand the drug addiction, use that as a paradigm to study the memory process. So for that purpose, the first thing we need to do is to classify the neuron type or subtype in this reward system and try to link that to identify the relevant cell type that mediates the addiction process. So what we did is classify the neuron type, then create mice for mark each of the cell classes. Then we try to manipulate, activate, or inhibit each of the neuron cluster and ask which one cluster actually can mediate the addiction process. Okay. After they identify the relevant cell type, then through neuron tracing to map the neural circuit, as well as then study the molecular change to understand the molecular mechanism that mediates the addictive process. So during the past 10 years, basically follow this trend, what we have done or what we have achieved to establish the relevant technique, including classify the neural subtype in the relevant reward system, PFC, nuclear contents of VTA, and general multiple occurrence, as well as establish the behavior paradigm for a conditional place preference, intervallous self-administration. That's so both are measure reward and addiction. We also establish cathode imaging and fiber photometers to monitor the neural activity, as well as the manipulation chemo and optogenetic tools to manipulate the neuron activity, as well as establish antegrade and retrograde viral tracing tools to study the neural circuit. Of course, we establish neural tagging and neural subtype purification method so that we can study the chromatin and epigenetic and transcription change. So during the process of establishing tools and profiling, we did publish 6 papers in the past several years that grouped into 3 groups. One's the profiling neuron subtypes, cell classification, and characterize the neuron, one specific neuron subtypes, as well as develop tools to isolate single neuron nuclei to do transcriptome and chromatin profiling. So today I'm going to briefly summarize 2 papers that we published several months ago in Nature Neuroscience, and as well as a paper that is now in press in Science Advance. Both are relevant to nuclear complex. So basically, this is the mouse brain. Now, the central part of the reward system is the prefrontal cortex nuclear complex, as well as VTA. So as the central part of the reward system, nuclear complex accept or receive signals or information from PFC, HIP, as well as the VTA. They also send signals to other brain regions. So basically, nuclear complex play an important role in multiple behaviors that I listed here, particularly relevant to my talk is reward and addictions. So basically, nuclear complex is quite complex in terms of projection as well as behaviors that it involves. However, the neuron compositions in this region is quite simple. So this is what people know several years ago before we start. So basically, they composed of interneuron as a middle spinal neurons. And middle spinal neuron, people know, is D1 and D2 neurons. And these are the classified according to the dopamine receptors, the D1 type and D2 type. So this simplicity in neuron type is so basically not consistent with the functional diversity as well as the neuron connective complexity. So how to reconcile this? So maybe the nuclear cell composition is not as simple as the D1, D2, and interneuron. So then we basically decided to classify the neuron type in this region use single cell approach. So basically we perform single cell analysis and identify different cell types in this nuclear complex. And particularly we identified 19 non-neuron cell type as well as 76 neuron subtypes. And particularly relevant to my talk is 13 interneuron type as well as 57 middle spinal neuron types. So in the 13 neuron type, we can show here, these are the 13 neuron type we identified, interneuron cell type we identified. The ones that marked with in green are the ones that people described before. As you can see, we identified many new subtypes of interneurons that are marked, some of them by single markers, some others you need to use combination of markers to identify them. And similarly, in middle spinal neuron D1, D2 type, we separately identified 30 and 27 subtypes of them. Okay, and we can also confirm use single cell RNA fish to confirm some of the new subtypes that we identified. For example, the SERPIN-B2 subtype here, these are the group in this single, so it's only less than 5% of the D1 cell type. You can see by cross-learning of no, by core RNA fish with the D1 and D2 marker, you can see it's D1 type, but not overlap with D2 marker. Similarly, TAC2 is another subtype that accounts for about 10% of the D1 neurons, and it's also belong to the D1 subtype and D1 subtype. And on the other hand, the Clary clusters, it's the overlap with the D2 subtype and with D2, but not the D1. So we can identify this. Now, this gave us some transcriptome-based cell classification. So the conclusion from this part is that single-cell RNA-seq revealed new interneuron subtype in nuclear complex. We also identified confirmed molecular distinct middle spinal neuron subtype in nuclear complex. However, what is the spatial distribution of these different neuron subtypes? So to answer these questions, we collaborate with Zhang's group using morphish to classify this different neuron type. First, we can classify the different cell type using morphish. And importantly, we can compare the morphish divided cell type and with single cell. As you can see, we can, these two identify cell type, they match very well. And importantly, this morphish allow us to look at their spatial distribution of the different subtype of D1 and D2. One important thing, as you can see very clearly, from anterior to posterior, different D1 and D2 subtype have different distribution. So meaning different, subtype, they are not uniformly distributed in the brain region, particularly in the nuclear compounds from anterior posterior. So in addition, morphage also allow us to reveal the potential regional specificity of cell types, the interactions of the different neuron subtypes. So for example, the TAC1 and subtype and the MPY2R subtype, you can clearly see that TAC1 neuron subtypes are mainly distributed in the shear and kind of depleted from core. On the other hand, the other subtype mainly enriched in core and not much in the shears. So similarly, we can find other neuron subtypes. Some of them, they have very similar distribution, suggests a possible functional link, while the others, like this gene as well as the receptor TRH receptor, they have similar distribution. Okay, so suggests they are functionally linked. So therefore, morphage allow us to mapping the transcriptionally distinct cell type in the mouse stratum. We identified different D1 and D2 subtypes. We also decoding the molecular and spatial features of middle spheroid neuron subtype underline functional complexity of the nuclear compounds. So the above studies so basically allow us set up the foundation to probe in the function of each of the subtype neurons that we identified. So what I'm going to tell you in the next 10 minutes or so about the TAC2 clusters that I mentioned earlier on, which account to about 10% of the D1 type indicated here. So, so basically this D1 type, this is the TAC2, which about 10%. And then on a fish indicate that TAC2 neuron mainly located in the medial shear as well as the core of the nuclear compounds. And importantly, we can use single molecular phage to confirm it's belong to D1 and not belong to D2. So now the question is this subtype of middle spheroid D1 neurons involved in cocaine reward or addictions? So to address this question, we set up two different techniques that allow us to manipulate the neural activity of these subclusters. One is the optogenetic approach. The other is chemogenetics, that you use chemicals based on the virus that you express either the receptors for HM3 or HM4, then you can either inhibit or activate the neuron subtype. So then this neuron activity manipulation is dependent on CRE expression so that we can achieve subtype group of neuron manipulation. Then we use a behavior test. In this particular one is conditional place preference, where we basically initially train the mouse to initially get the conditioning. So on one side of the cage, then we will give the cocaine injection, where the other is just saline injection, so that they are conditioned. Then at day 4, we basically put the mice, let the mice to choose which side they want to go. Okay, so this is a condition place preference test. So we basically ask when we activate or inhibit the neuron of this subtype, then what's the influence on the choice of the site by the mice? So in the control, as you can see, after the training or conditioning, the mice preferentially go to the site that have cocaine injection. So as you can see, this pre and post conditioning, they have the difference. However, when we activate this neuron cluster, then this difference is kind of blunted. So indicating activation of these subclusters will favor will suppress the cocaine effect or addiction. So this is quite surprising because the conventional view of the D1, D2 type is like this. So basically, D1 is enhance the cocaine behavior or induction, where the D2 is suppressed. So our analysis of part of the D1, subclass of D1, actually the opposite is inhibit when we activate. So therefore, we want to use additional ways to confirm this. Now we switch to use chemogenetics. So we inject this mice with the AAA virus that either express 3DQ, that is activate. Okay. So as you can see, similar to genetic result, activation of this neuron cluster, basically kind of inhibit. Okay. On the other hand, when we inject the M4D, which is the inhibit. So when we inhibit, now we see from the control that show no difference. Now this is at a different dose of cocaine. So then we can see that increase this. So basically we can conclude, and then, so the CPP is measure reward. So therefore we want to know, whether it's involved in addiction. So addiction needs self-administration, so self-promoting process. So therefore we use the cocaine IVSA paradigm or behavior test. So in this test, so we basically connect this tubing that is involved, you can get a drug to the mouse blood vessel. So when mouse push the active level, they will get the infusion. On the other hand, this is coupled with the Q legs. On the other hand, the inactive level, when they push, so there will be no drug induction. So we basically can't, which level the mouse will push. Based on this, to measure their addiction levels. So then after training of the mice, we randomize the dose that the mouse will get, then measure the dosage responsible curve. So what I show you here, as you can see, compare with the control activation using chemogenetic activation of that clusters, you can see that reduce the cocaine self-administration. So this is the level press, this is the level press accuracy, and this is the infusion total the cumulative infusion. As you can see, activation of these clusters in all these measures is decreased. On the other hand, when we use inhibit these neuron clusters, then all the measures indicate that the inhibition that increase the cocaine taken. So based on this result, we can conclude activation of the TAC2 D1 neurons negatively regulate cocaine reward, while on the other hand, repression of these TAC2 clusters positively regulates the reward. So similarly, in the addiction assay, in the IVSA assay, we can see the similar result. So next we ask, how do the TAC2 neuron intervene and elevate the downstream target that mediate these functions? So for this purpose, we traced the projection of this neuron type subtype. We found that the injection in the nuclear comments, then we can find this neuron project VP project to native hypothalamus as well as VTA. Okay. Then we basically use optogenetics to stimulate the terminal each of these projections and then measure their effect on the CPP score. So the cocaine, the condition place preference, we only found that the projection from nuclear compass native hypothalamus actually is this group that responsible for this behavior change when activate this case, this group of neuron. So we can conclude that the TAC2 positive neuron that projected to 3 brain regions, and it's the projection to natural hypothalamus that mediate this cocaine reward behavior. So in summary, so basically I told you 2 stories. One is that we constructed a spatial resolved cell taxonomy that for the understanding of structure and function of the nuclear complex. Then we identified and conventional rule of the molecular defined D1 subpopulation, the TAC2 neuron, that in suppressing the cocaine reward and addiction, which is which is different from the conventional view of the whole D1 neuron that positively regulate cocaine addiction. So the people who contributed work, so the taxonomy, the neuron subtype is mainly the work of former postdoc Rinchao, and the data analysis is carried by former postdoc Nadia. And the second story mainly is the work of the Chen Dong, and the other people in the lab that also contributed to this work, including former postdoc Xiao Yichong, current postdoc Yichong, Ritra, and former postdoc Wen Chang. And we collaborated with Xiaowei's group in Morefish, and Qiufu actually kind of provided us with the TAC2 creamice, and the work is supported by NIH and the Hughes. Actually, I want to take this opportunity to advertise postdoc positions. So in addition to the pre-implantation development and drug addiction and pain relief drugs that are going on in the lab, actually we also have other projects, including the one that they basically in study the extra chromosome circular DNA, we revert its link to innate immunity. So therefore we have a small group, try to study the native immunity. We also get funding and we initiate a project, try to study the role of the tissue stem cell in aging from the epigenetic point of view. So if anybody have interest in these directions, you're welcome to email me. We can see whether our interest is fatal or not. So basically, that's my talk. Thank you very much for your attention. Now I will stop sharing and answer questions. All right.
Yep. Cool. Thank you, Yi. This is a very interesting talk, and we got several questions. So Li Jun have questions for you. Has questions for you. So amazing heterogeneity you described for nuclear compound cell types from their spatial distribution. Do you think they represent discrete cell types or continuous variations of a cell subtype, super types?
So it's a good question. Clearly, you can see we showed different subgroup that show a kind of gradient, or actually in some part they don't even exist. So I would say it's a combination. There are some places you show a gradual change. There are some subgroup that really just enriched in some local brain regions from anterior to posterior. So it depends which subtype of neuron you're talking about. It could be both ways.
Mm-hmm. And I have a quick question, because there are co-expressing D1, D2 neurons, right? So when you do this subtype screening, do you find like any specific, like for example, for the D1, D2 co-expressing cells, they have some unique genetic profile, or any specific things you can like use like if we have a like new target to label those D1, D2 co-expressing cells?
So actually, when we so this is a question that when we publish, the people also ask. Indeed, actually, the overlap is very few, very few. So the initial D1, D2 separation of different cell neuron type, actually, that's quite good. So we found very few that express both D1, D2. Actually, later on, we do further analysis, and we believe those one might be a double. So basically, sometimes the single cell analysis you can get beads that can have contamination. Okay, so in general, I would say very few neuron that really express both D1, D2. Yeah.
Mm-hmm. I see. Cool. Thank you. And then we have the more questions. I'll just select two, and then we have to move on. One question is about is the diversity of neuron subtype in neoclicampus dynamically changed during the development of drug addiction?
So that's a good question. So we have not done the work that to analyze different phase of the different subtype. But I will say, it's possible that during neuron development, like from young to old, that dynamic change. But it's hard for me to imagine that you have subtype change. So basically, subtype change requires new neuron generation, so right, or change cell phase, change cell subtype. I do not think that the process, addictive process, can make such a dramatic change, because we did single cell transcription analysis and compare different phases of the addiction, but we do not see such a dramatic change so that they can be classified into different group.
Mm-hmm. Yeah, and then Shmuel has a question about very general. I think the question is to ask what can be the contribution of unique genes of everyone to the level and intensity of addiction to drugs? I guess like individual levels are different. Is any genetic contribution there? It's I think.
So that's an interesting question. So basically, if you do the self-administration test, you will see even the mouse, some will see very addictive, some less. So given genetically, they are the same, right? So people believe maybe some other thing will affect, so the question is, it's very difficult to try to figure out what are the difference between different individual mouse, because there's so many things you can look for. Well, first of all, if we don't know what cell, what neuron type to look, not to mention that even the same neuron, same a mouse, what other modification you're going to look, what gene, where. So bottom line, at this point of view, even we want to study, I don't think we have a way to figure that out easily. Okay.
Yeah. Cool, great. Thank you again, Yi. So this is really wonderful and we've learned a lot. So we move on to our third talk. Qiufu is going to present his work. I'll give a brief introduction. Dr. Ma received his bachelor degree from Fudan University in 1987 and the PhD from UCLA in 1994. After postdoctoral training with Dr. David Anderson at Caltech, he joined Harvard Neurobiology Department and the Dana-Farber Cancer Research Institute in 1999 and became a full professor in 2011. His lab has been working on the somatic sensory circuits from neuronal subtype specification to spinal pain circuit mapping. Most recently, his lab started to map somatosensory autonomic circuits as a way to understand how acupuncture works. Today, he will discuss a neuroanatomical basic for acupuncture to modulate systemic inflammation. Let's welcome Qiufu, and we look forward to learn about the acupuncture in the neuroscience space.
Yeah, and I would like to thank Nam for the nice introduction, and also thanks HMS-CSSA for this opportunity. So in this afternoon, I'm going to discuss some of our recent work regarding how acupuncture can modulate inflammation by driving some specific neuronal pathways. So for this audience, we all know that acupuncture has been practiced for a long, long time in China, but also now in all over the world. And the United States has 30,000, licensed acupuncture doctors. In Dana-Farber here, have 5 acupuncture doctors treating those side effects, actually associated with cancer patients. So based on the clinical trial, study, WHO now have recommend, almost over 100 medical conditions for acupuncture, practice. And many of those conditions is chronic disease problem, which has a lack of good treatment from Western medicine. But despite this very wide use, acupuncture as a scientific basis behind this practice is still not well understood. So, and I see a lot of debate for the field as well. So if you talk about acupuncture, and we have to mention the meridian channel hypothesis, which forms a co-foundation for how acupuncture to work. So according to this hypothesis, all body parts has been functionally connected. And at the ancient time, they thought through those meridian channel. And this functional connectivity allow an acupuncture stimulation at a distal body region, like a toe or hind leg, to distantly modulate body physiology, like a gastrointestinal motility, for example. But this, so for many decades, study people in China, Korea, and Japan try to understand, what's the nature of this, meridian channel hypothesis. So any scientific, basis, and then no one, can really describe or can find any kind of physical, like a channel, make this kind of connection along those meridians. So this kind of lack of those physical evidence is cause a lot of doubts, debate for the field, both in China and the United States. For example, many people in the Western research community think acupuncture is just somehow produce a strong placebo effect and no real scientific basis. And even in China, there's a lot of doubts as well. So I remember a few years ago, I attended a committee meeting in Shanghai, and after one acupuncture presentation, the former president of Shanghai Institute of Biochemistry, the institute I came from, and he makes this blunt comments. He say, anyone who believes in acupuncture is not a serious scientist. At that time, we just started to think about doing acupuncture. So so here, today I'm going to talk about the somasensory autonomic reflex. So I will share the idea that this kind of reflex does allow acupuncture to modulate body physiology at a distant size. So we know that autonomic nervous system, like including sympathetic neuron and the parasympathetic neuron, virtually control almost every aspect of our body physiology, and including inflammation modulation, which I'm talking about today. And my lab actually for many decades, 2 decades, at Hafler here is actually studied the somasensory neuron, and this neuron crucial for us to sense pain or itch or touch or temperature. We all know that activation somasensory neuron has a crosstalk, can generate crosstalk with autonomic nervous system. For example, any powerful injury stimulation, if you cause intense pain, you're going to increase your heart rate, for example, by increasing sympathetic neuron activity. And after the stress removed, and if you have a body injury and the parasympathetic nervous system are going to be activated to reduce your activity and then to promote a wound healing. So this kind of somasensory and autonomic reflex, which it can carry some of the, conceptually can carry those meridian channel hypothesis, meaning that acupuncture stimulation in the somatic tissue can distantly modulate body physiology by driving this kind of autonomic nervous system. So about like 6-7 years ago, we started to think about doing this kind of scientific basis behind acupuncture. So the basic approach we use, we started a pain study actually, we try to use a genetic tool to manipulate the somasensory neuron and autonomic neuron, like for example, to ablate, silence, or activate those sensory autonomic neuron. Initially, we studied the impact on pain sensation, but in recent years, we want to study how this kind of sensory autonomic neuron can impact acupuncture effect. So then we are visiting a bunch of places in China, talking to a bunch of acupuncture scientists and doctors as well, to looking for what's a kind of robust, model system to study acupuncture in animal, like in mice. So one of the model systems they introduced is called the modulation of the severe cytokine storm by acupuncture. And during this pandemic time, I think everyone knows that this cytokine release syndrome is one of the major reasons cause a high fatality for those COVID-19 patients. And in the United States, we have 1 to 3 million of the patients annually, even before the pandemic, with a very high fatality rate, 15 to 30%, and still no FDA-approved drug to treat those severe cytokine storm. But pioneered by Kevin Tracy and many others in acupuncture field now shown that like vagal nerve stimulation and later on by acupuncture can drive those kind of autonomic pathway, can powerfully shut down those cytokine storm, greatly promote animal survival in animal. So we borrowed this experimental system to study the neural anatomic basis, how acupuncture really works. So we started the work in about 5 years ago, and then now with this first 2 papers we published, we've shown that there is indeed some kind of interesting organization rule for acupuncture to work. So there's many factors can impact on acupuncture effect. And in today's talk, I'm only touched the one aspect called the body region specificity. Or you can say core idea behind acupuncture practice. Is there any scientific basis behind acupoint specificity or selectivity? So this pathway I'm going to mention is called a vagal adrenal anti-inflammatory axis, which is initially discovered by Louis Ula. Now he's a professor at Duke, published a landmark paper in Nature Medicine. And Louis Ula lab, found that acupuncture stimulation in the hindling region can drive this axis, starting by activating somasensory neuron. The signal will release through spinal cord to the hindbrain, eventually leading to the activation of the parasympathetic efferent neuron, and then cause the adrenal gland to release as a catecholamine like dopamine. And this dopamine release can powerfully reduce systemic inflammation induced by bacterial endotoxin like lipopolysaccharides. And what we found that this pathway actually interesting, can only activate from the hinding region, but not from abdominal region. So first we repeat, the experiment done by Louis Wu and many other lab, by performing electrical acupuncture in the Zhusanli, the hinding region, by inserting the needle through the anterior tibialis muscle deep into a region close to the bone and nearby to the two major nerve called a deep like a peroneal nerve. And by electrical stimulation 15 minutes in a low intensity, at an intensity likely is not painful, was sufficient to activate those parasympathetic efferent neuron located in the hindbrain, like indicated by the C-force induction. And then we shown that, we shown that this acupuncture, can reduce a systemic, inflammation or cytokine release, in response to the LPS exposure. And this reduction of the acupuncture effect, in terms of anti-inflammatory effect, it depends on vagus nerve. If you cut the vagus nerve, and this acupuncture effect will be lost. And we then show that a group's chromatin cell located in the adrenal gland marked by the neuropeptide expression NPY. If we remove this group catecholamine neuron, then the acupuncture effect will be also lost. And then we shown that this vagal adrenal axis has a body region selectivity. If we do the same stimulation in abdominal region, you cannot produce anti-inflammatory effect. Even we use a very high intensity electrical stimulation, if we cannot induce C-force in those vagal, efferent neuron. So this is a kind of remarkable, you can see the body region selectivity, or you can say some kind of acupoint selectivity. The vagal adrenal axis can only evoke from the hinding region, but not from abdominal region. Then the question became the anatomical question, say why, why the vagal adrenal reflex can only be evoked from the hinding region, but not from abdominal. So then we can thinking about it, so there must be some kind of sensory neuron, maybe innervate tissue very unique to the limb region, like it has all kind of joint, ligament, and a large skeletal muscle or bone, which you do not have in the abdominal region. So this is our hypothesis. We're looking for those neurons. And this, to help at the searching for those neurons, is build our earlier developmental study. We know that the somatosensory system actually can divide into two major branches. One branch go to skin epidermis, or like a defensive reaction for animal to avoid external threats. So there's another branch sensory neuron with monitor your body integrity, which innervates throughout your body, including those limb, like a deep fascia tissue. So we engage almost half dozen screen line, eventually identify a group sensory neuron, and which we now shown that are crucial for driving the vagal reflex. And this group neuron only label about it like a subset of sensory neuron, which can show the expression nerve growth factor receptor like TrkA. Many of them are myelinated and a peptidogen neuron. And what's the interesting part, cause to encourage us for first study is we can see the differential distribution of this group neuron along different axes. For example, this neuron are highly engaged in the sensory ganglia at the limb region, but not in the like forelimb or hindlimb, but not in the thoracic region. In terms of percentage-wise, have 3-4-fold difference. And then we look at the innovation about this group neuron. And I want to highlight, if you talk to acupuncture people, they always mention, in the traditional old time, we always stimulate some kind of like a fascia tissue, Tumor. This is our network of fascia tissue. If you remove the bone and the muscle, and this is the whole body wide, fascia tissue wrap every bone and the muscle joint, and this is body tissue. But we found that this fascia organization is it's really showing the also tissue, body region selectivity. For example, we found this group sensing neuron innervates the densely innervates the fascia in the limb region, like a periosteum, ligament, joint, and a plantar fascia, and all those limb region, but not innervates the fascia tissue within the abdominal region. And this actually really first time showing the differential sensory innervation to the different part of the, our body fascia tissue. And then we saw this could be a great candidate for sensory neuron for acupuncture drives the vagal reflex only from the limb region, not from the abdominal region. So we use a genetic tool to selectively to activate or to ablate those sensory neuron only within those sensory ganglia. And then we test acupuncture effect. And you can see that without after ablating those group sensory neurons, the acupuncture effects are lost in terms of driving the vagal adrenal axis to produce an anti-inflammatory effect. And also when we did as a optogenetic study, to introduce a channel rhodopsin into this group neuron and insert the optic fiber into the hinding region at a ST36 site, and it can mimic acupuncture effect, can drive the vagal reflex, can produce an anti-inflammatory effect in a vagal nerve dependent manner. And it can greatly promote, animal survival. Animals with the dose of the LPS we use, you can increase survival rate from 30% to almost to 80%. And this just by 15 minutes electrical acupuncture. It's almost too good to be true, but that's repeated by many different labs. And in terms of acupuncture, same thing. So then also what we found interesting, and this acupuncture or this group sensory neuron innovation pattern can really help us to retrospectively to predict what body region will be effective or not effective. For example, I told you that this group neuron also enriched in a folding region, not just a hinding region. So we insert a needle close to the one of the deeper branch of the radial nerve, which innervates the inner bone periosteum, can also produce anti-inflammatory effect in a vagal nerve dependent and a procreative neuron dependent manner. And this is one of the, I think, one of the more impressive funding which will give us to appreciation the Asian wisdom. We don't have to believe all those acupuncture selectivity along our body region joined by those meridian channels. But if you look at the hinding region, you will appreciate, there is a meridian channel, along the midline, and this is right underneath this line is almost precisely follow the sural nerve. Right underneath this line is a sural nerve. Okay. But in each side, the middle line is a big muscle like gastrocnemius muscle or semitendinosus muscle in the side region, which do not have a traditional acupoint and often use a non-acupoint control in acupuncture study. And if you look at the nerve distribution of a neuron, compare the inner compound muscle in the anterior tibialis muscle from anterior side, the Zhusani. And if you look at the gastrocnemius muscle in the posterior side, the density of the sensory fiber innervation almost a tenfold difference. And if you insert same depth needling in this big muscle, you cannot produce acupuncture effect. And it's the same thing in the side region, like semitendinosus muscle. And we also, I didn't show you data within the anterior tibialis muscles, it's an inner component, outer compartment. The outer compartment is more like a gastrocnemius muscle. So our nerve is only in the inner compartment, but not as outer compartment. If you Zhusani, if you put needle in the superficial, you also cannot produce anti-inflammatory effect. So this study is to give you a flavor, the proof of principles that there is some kind of anatomic organization for acupuncture to drive, specific autonomic pathway. And this acupuncture selectivity or specificity, partly, explained by the distribution of sensory sensory nerve has a differential, amazing organization within our body. So what I told you today is basically is kind of like showing it's really, you know, there's some kind of, I just mentioned, scientific basis behind acupuncture. We all know that somatosensory systems and autonomic nervous systems are fairly complex. And this crosstalk is a complex system crosstalk. You need a central nervous system integration, and all the target tissue have to listen to all the different autonomic pathways. And it's really can open up many system physiology level study in the future. And if you think about it, you know, all the type of human chronic disease, which we do not have a good solution from Western medicine, inflammation is always one of the, could be the components. I think by study those nerve immune target tissue interaction, I think, and particularly can use acupuncture or any kind of physical tool to mobilize this kind of interaction, could open new way to study or even to treat those many forms of human chronic disease. So this is really a wonderful collaboration between USA and China. This is the first generation. This is the visiting student and scholar from Fudan Beijing Institute, you know, acupuncture and Moscow Bastion and Fujian Medical, Fujian University Traditional Chinese Medicine. And with a student or former postdoc, Yanqing and Xiang Hong. And this is the people, you know, come to my lab to set acupuncture study. And at a time when we do have more difficult to do the collaboration between the US and China, and this is such a great example showing that Western, you know, USA, China collaboration can do something great. So I will stop here, you know, and well, happy to answer any question. Thank you.
Thank you, Qiufu. It's very interesting. So I have a question before I open to others. This is like my priority as a moderator. I'm just curious, like, right? So when you do the mouse model, how do you decide, like, because human acupuncture, how do you know when you electrically stimulate there, although you show the anatomical structure of the neuron and muscles, and how do you know when you're doing in the animal, it is the acupuncture you are doing in the human?
Yeah, and yeah, so the answer, that's, you know, so the question is that we always face, because true, we do not have enough resolution to like a small animal like mice, but still, what we touch here is some kind of hierarchical organization, mean that some of the larger difference, like in this case, serous sensory neuron, like has differential distributed to the cutaneous or deep fascia, and a serous sensory neuron has an organized in a rostrocaudal, you know, direction. So we only touch the larger scale, you know, difference at this moment in animal. But even with that, you know, the hinding region, like small animal like mice, like we always see the anterior side, posterior side different. Even within the ST36, the depth of the needling, we only use 3 millimeters into the muscle, but we still can see the different by shallow or the deep, you know, stimulation. Mean that the focal electrical stimulation has some resolution already, allow us to explore some anatomical difference. But a fine detail, I think, you know, human will be, you know, large animal will be better resolution. I think that the basic larger organization should be evolutionarily conserved. So, okay, yeah.
And okay, so there's a question from the audience. It's about what do you think about this 学道? Is it just neuron? I think it's a relevant question also.
Yeah, it's here I think that we only touch the long distance effect, you know, acupuncture, and the, you know, mean that how the acupuncture can changes the physical organ or like an adrenal gland. So this kind of like a long distance effect in some way have to involve the nervous system or neuroendocrine system, and but in terms of physical stimulation within the acupoint, of course, it's a very complex. So you have injury, and you have mechanical sensitivity, and so you have secondary dynamic interaction, like when the needle deep, like you causing tissue injury, and as the injury tissue will release those chemical mediate, can activate many different from the sensory neuron, and also non-neural tissue as well, like also mast cell or target tissue. So it's a dynamic interaction between the the nerve, immune, and the target tissue within the stimulation site. And how the acupuncture can locally change the disease, like when you have a joint pain. I think that's a whole different biology needed to study. Yeah.
Yeah. Great. Yeah, I think you also answered the other audience questions. And one last question is about have you like the anti-inflammation, all those biomarkers, for example, you have found this in the serum, they have TNF alpha levels changes. Do you have like human subject this similar kind of measurements for acupuncture or?
Yeah, I didn't go to very detailed, truly, in our case, acupuncture stimulation in ST36, for example, you can enough to cause an increase of the catecholamine, like dopamine or adrenaline level within the serum. So you can measure, take the blood to see the significant increase. And then you can measure the changes, the cytokine level within the serum, because we use the systemic inflammation as a model system. And the human study, definitely, eventually we had to bring back to the human. For example, we wish we can do some human study in the future. But as a field, like my collaborator, Professor Bingzhu, talk about the gastrointestinal modulation by acupuncture. They clearly see a difference both in animal and human when you stimulate limb region versus abdominal region can cause opposite, enhancing or inhibiting gastric motility, showing that there is some kind of acupoint selectivity and consistent with our driving different autonomic pathways. So, but I say that's one of the future, definitely important. Crosstalk, we can borrow a lot of human experience, like a human experiment for 1,000 years. Those can really help for us to form the hypothesis and entry point. But eventually, our study can really also bring back to improve the acupuncture practice. For example, I didn't show you like if you to treat the cytokine release syndrome, not to prevent, and it's a stimulation pattern is so important. If you low intensity in Zhang, for example, can drive the vagal adrenal, can reduce inflammation, promote animal survival. But if you give it high intensity, can drive the sympathetic pathway, can promote inflammation, can kill the animal quicker. So they're showing that, and I talked to a bunch of acupuncture field, and a few people recognize this kind of like detrimental effect by acupuncture. So I think there's a lot of room for crosstalk between animal study and human study. Yeah.
Awesome, great. Okay, so I think we are running very out of time, and I want to thank again Qiufu, and then thanks to our other speakers in this session, Li Chun and Yi. This is really a wonderful morning. People, so just feel free to contact them if you have more questions, and I'm really happy that we spent this wonderful morning, Saturday morning here.
Thank you very much. Yeah. Bye.
Thank you, Dr. Nally, for helping us moderating this session. Thanks to all the speakers. Now we are able to have a lunch break. Please come back at 1:40 Eastern Time for an afternoon sessions about structural biology. We will first have a ceremony for HCLS Biocytogen Distinguished Research Award before the session 3. So the chat box, we will make it available during the break. Thanks. Please come back at 1:40 p.m. Dan, would you like to share the slides for our association?
Yes, give me a second.
Hello, Dr. Tammy Fu. Thanks for coming to help us moderate session 3. So in a while, we would first have a award ceremony, and at 2, we will start session 3. Thanks.
Hi, E. I think we're gonna start the ceremony in like 1 second. Do you mind stop sharing your slides? I can pull up my slides.
Oh yeah. So sure.
Okay, it's been 1:40 p.m. and welcome everybody from coming back from the lunch break. And it's my honor to host the coming session, which is the 2022 HCLS Biocytogen Distinguished Research Award Ceremony. And really thanks to one of our diamond sponsored by Cytogen for sponsoring this event. And also congratulations to all the awardees for this outstanding award. So every year at the symposium, we will also receive applications for this award, mainly among outstanding young researchers around the HMS community. And it's really exciting to see that all of the awardees for this year are able to join us on Zoom today. And what we're going to do is I'm going to share the slides, and I will call their names one by one, and each of them will turn on their camera and audio and speak 1 or 2 sentences about themselves. For the sake of time, please make sure to be aware of that. So I'm going to start with our first awardee for the Distinguished Research Award. It's presented to Dr. Yingzi Yang from MIT. Dr. Zhang, do you want to talk to us a little bit about yourself?
Yeah. Good afternoon, everyone. My name is Yingzi Yang. I'm a postdoc fellow in Guoping Feng's lab and currently in job market. My postdoc work focused on 2 understudied thalamic nuclei, anterior thalamus and perivascular thalamus, and their role in neurological disorders. My long-term vision is to develop therapeutic approaches, starting with animal models that could one day help human patients. I feel so honored to receive this award, and this would not be possible without the great help from my postdoc mentor, Guoping Feng, as well as my collaborator, Diraj Roy. Many thanks to the HMS-CSSA organization and the award committee. Best wishes to all of you. Thank you.
Thank you so much, Yin. And we're going to move to the next awardee, Dr. Rongqing Pan from Dana-Farber at Harvard Medical School. Dr. Pan.
Hi, everyone. Good afternoon, everyone. I'm Rongqing Pan. I got my PhD degree from the University of Texas MD Anderson Cancer Center, working on cancer-targeted therapies and drug resistance. My PhD work was published in Cancer Cell and Cancer Discovery, and most importantly, this work paved the way for 2 clinical trials and led to FDA approval of 1 drug called venetoclax to treat AML. During my PhD training, I identified several intriguing intersections between cell dyes and immunotherapy. Then I joined the Anthony Latta Lab and Dana-Farber to explore these ideas. And currently, I'm working on how to effectively augment NK or T cell-based immunotherapies and how to overcome drug resistance by targeting cell death, especially targeting the mitochondrial apoptosis pathway. I feel very honored to be selected for the Harvard Chinese Life Science Distinguished Research Award, and many thanks to the HMS-CSSA for organizing such an amazing event. I hope you all enjoy the rest of the conference and the long weekend. Thank you.
Thank you so much, Rongqing. Next we have Dr. Yuangao Wang from Boston Children's Hospital at Harvard Medical School. Yuangao, are you there? Yeah, I think you probably need to unmute yourself. Yuan Gao, I think I can see your audio, but I could not hear you. So sorry, maybe for the sake of time, I think you're trying to connect to the audio right now. Maybe for the sake of time, let's move to the next awardee and we'll come back to Yuan Gao maybe in a second. Okay. Okay, next, oops, sorry. Next, we have Dr. Jun Zhang from Boston Children's Hospital at Harvard Medical School.
Good afternoon, everyone. I'm Jun Zhang, and currently I'm a postdoctor in Professor Bing Chen's lab at Boston Children's Hospital. My postdoctoral research focuses on the studying biochemical structure and function of the viral fusion procedure for several types of enveloped viruses, including the HIV, SARS-CoV-2, and related viruses. It's my great honor to have this award. Many thanks to the HMS-CSSA community and also for the award support community. Best wishes to all of you and have a great weekend.
Thank you so much, Jun. Next, we have Dr. Miao Gui from Harvard Medical School.
Hello, everyone. I'm Miao Gui. I'm a postdoc in the Alan Brown Lab at the Department of BCMP at Harvard Medical School. My research, I'm interested in using cryo-EM to study the structure of cilium protrusion at the cell surface, which can help the cell to move or sense the outside environment. I'm also interested in understanding the mechanism of cilia-related diseases. It's my great honor to receive this award. Thank HMS-CSSA and the award committee. Thank you.
Thank you, Miao. And next we have Dr. Haining Zhou from HHMI and Department of Cell Biology at Harvard Medical School.
Hello, everyone. Hi, my name is Haining Zhou. I'm now a postdoc from Dr. Chemogenetics lab in Department of Cell Biology. So my research work is focused on epigenetic gene regulation mechanisms. Recently, we found a new nascent RNA degradation pathway that mediates nascent RNA degradation protein that mediates gene repression. Today, I'm so honored to receive this award. Thanks to HMS-CSSA for organizing the event. Thanks to sponsors, and also thanks to award reviewers for recognizing my research work. Thank you.
Thank you, Haining. Next, we have Dr. Yancong Zhang from Harvard Chan School of Public Health and Broad Institute.
Hello, everyone. My name is Yancong Zhang, a postdoc from the Harvard Lab at the School of Public Health. My research interests are focusing on understanding the function of the microbiome community, particularly that of the human microbiome. That means utilizing computational biology, molecular biology, and biochemistry to study their potential impact on human health and disease and improve healthcare outcomes. It's really my great honor to get this award. Many thanks to all the organizers and the committees. Nice to meet you. Thank you.
Thank you, Yancong. Next, we have Dr. Pei Qiangfeng from Harvard Medical School.
Hello, everybody. My name is Pei Qiangfeng. I'm currently a postdoc fellow in the lab of Tumor at the Department of Cell Biology, Harvard Medical School. And my research mainly focuses on the mechanism of protein translocation into peroxisomes, which is known as the essential organelle of all eukaryotic cells. It's really my great honor to receive this award, I'm grateful to all committee members and organizers to bring me in this great community. And I also want to take this opportunity to thank to all the people who helped me during my postdoc career, particularly my mentor, Dr. Tumor Hafler, and all my collaborators, colleagues, and also my families. And I wish you everyone enjoy all the sessions of this great meeting today, and you have a great weekend. Thank you.
Thank you, Pei Chang. Next, we have Dr. Xiaozhe Xiong from Boston Children's Hospital.
Hi, I'm Xiaozhe, a postdoc from Dr. Mingdong's lab at Boston Children's Hospital. We are focused on identifying bacterial toxins and their function in pathogenesis process. It's my great honor to receive such important award. Winning this award would not have been possible without the inspiration I have received from my supervisor and collaborators. I also would like to thank the organizer. I really appreciate it. Thank you.
Thank you, Xiaozhe. And just to let everybody know, we will hear a awardee short talk from Xiaozhe towards the end of the symposium. So please stay tuned for that. And next we have Dr. Pei Tong from Brigham and Women's Hospital, Harvard Medical School.
Hi, everyone. My name is Pei Tong. I'm an instructor of doing Wisman Lab at Brigham and Women's Hospital. I got my PhD from Tsinghua University studying HIV virus. The long-standing problem how HIV broad neutralizing antibody is induced inspired me to study the basic B cell biology. And my focus has been on understanding memory B cell recognition and revolution to foreign antigens. I'm going to join Hefei Comprehensive National Science Center Institute of Health and Medicine in August as a principal investigator to continue my B cell biology study. Thank HMS-CSSA community and award committee. I hope you all stay safe and enjoy science.
Thank you, Pei Tong, and really congratulations to your new position. And I think we're going to come back to Dr. Yuangao Wang a second ago. I think
Can you hear me now?
Yes, yes.
So I'm sorry. Hi, good afternoon. My name is Yuangao Wang, a postdoc from Yingzi's lab at Boston Children's Hospital. We focus on studying actual chromosome circular DNA. We clarify the genetic origins, biogenesis of ECC DNA. We found that ECC DNA exists strong immunostimulatory activity, and we're trying to identify its role in vivo now. Many thanks to the organizers of the CSSA. It's my honor to have this order. Best wishes to all of you, and hope you join the rest of this meeting.
Thank you, Yuangao, so much. So here is all the 10 awardees for this year's HCLS Distinguished Research Award. Thank you again for our sponsors, and also big congratulations to all the awardees for their outstanding research. And I think I'm going to stop sharing my slides and give the podium back to Yi.
Thank you, Dan. Congratulations all the recipients. It was a really competitive awards. So let's kick off session 3 now. So for this session, we will have also 3 speakers. Dr. Hao Wu as our keynote speaker. She's from Harvard and Boston Children's Hospital. Follow her will be Dr. Eric Gu and Dr. Alon Ke. So we have our moderator, Dr. Tianming Fu here. So let me briefly introduce him. Dr. Tianming Fu earned his PhD in biochemistry and molecular biology from Peking University in 2011. He did his postdoc training with Professor Hao Wu at Boston Children's Hospital and Harvard Medical School. He started his own lab at Ohio State University in 2020, and he has been focusing on mechanistic understanding of lysosomal signaling and immune signaling using biochemical and biophysical approaches. Thank you, Dr. Fu. You may now, I see your camera turn on and audio turn on. It's your stage now. Thank you.
Thank you for the very nice introduction. So we are still a little bit early. Should we wait a few minutes or should we start now?
I think Dr. Hao Wu is here. We can start because in the morning we took a lot of time and we didn't we didn't have the morning break, so I think it will be a very great discussions and talks.
Okay, so let me.
I'll be on time.
Yeah, let me introduce our first speaker in this session, Professor Hao Wu, my beloved mentor. Hao received her PhD from Purdue University under the supervision of Professor Michael Rossman. Then she moved to New York City and worked with Professor Wayne Hendrickson as a postdoc fellow. In 1997, she joined Weill Cornell Medical College as an assistant professor and was promoted to full professor in 2003. In 2012, Hao relocated to Boston Children's Hospital and Harvard Medical School as the Asha and Patricia Springer Professor. Hao has made exceptional contributions to our understanding of immune signaling. Collectively, her work established a new paradigm in cell signaling that is mediated by high-order assemblies, also known as signalusome. Hao was elected as a member of the U.S. National Academy of Science and the American Academy of Arts and Sciences. Additionally, Hao is also an exceptional mentor who always sets a very high standard bar for everyone in the lab. And she takes care of all the trainees' future. So with that, we are looking forward for Hao's talk on inflammasome today. Hao, you can take it.
Thank you, Tianmin, for that very generous introduction. We really miss you, Tianmin. Can you see my slide?
Yes.
Yes, great. So let me also thank the CSSA leadership for organizing such a wonderful annual symposium. I would also like to congratulate all the distinguished science awardees. And I'm also honored to share stage with Eric Gros and Ailong Ke. So here's my disclosure. My lab has been interested in using structural biology to elucidate fundamental questions in immunity. And as Arlene Sharpe has said this morning, that this is a great time for immunology, both for basic science and translational research. So the immunity that my lab has been mostly focused on has been the innate part of the immune response, which offers the first line of defense against infections and other types of danger. So innate immunity relies on so-called germline encoded receptors. And these receptors are present in barrier cells, such as skin and the epithelia, as well as immune cells of the myeloid lineage, as shown here, macrophages, monocytes, and dendritic cells, for example. By contrast, adaptive immunity is much slower, and it relies on gene recombination and clonal selection to derive highly specific T and B cells to combat infection and other danger. So inflammasome is part of the innate immune system. And I usually like to divide the innate immune receptors into two different classes: those that sense danger signals outside the cell or in the endosome, such as the toll-like receptor, and those that sense signals inside the cell, that is, when the danger actually breached the wall of the cell, and those are much more dangerous situations. And as one says that drastic times require drastic measures. So, and this is essentially what the inflammasomes do. That is, it not only induces cytokine secretion, but oftentimes induces cell death, which I think is the ultimate measure of defense. So inflammasomes contain receptors of what I call two different kinds. One is for direct recognizing
patterns in bacteria or viruses or other types of dangerous situations, bacterial lipopolysaccharide, for example, or double-stranded DNA or double-stranded RNA in the cytosol, and that's where they don't really belong. So these are direct sensing receptors, but there's also a lot of innate immune receptors that are not really a classical receptor. They don't recognize something specifically, but rather recognize some kind of a disturbance or effects from, for example, pathogenic bacteria or viruses. And this can also distinguish between commensals versus pathogenic bugs. So inflammasomes are supramolecular complexes that activate caspase-1. And the first inflammasome was identified in 2002, really after a very long crusade to try to understand what is the molecular basis of fever. So at some point in history, it was realized that fever-producing activities is mediated by proteins because they're heat sensitive. And these then, after decades of research, led to the identification of interleukin-1, a pro-cytokine it turned out to be, which means you would need something else to activate the cytokine. And that led to the discovery of the first caspase-1. And then inflammasome, of course, is the molecular entity that will induce the activation of caspase-1. So we now know a lot more about inflammasomes. There are quite a few different kinds of inflammasomes, but most of them are induced, at least initiated, by proteins in the so-called NLR family. This is an acronym, nucleotide binding domain containing and leucine-rich repeat containing proteins. So many of them belong to this same family. And today I will focus on NLRP3. So most of these inflammasome proteins, upon stimulation, would recruit this adapter known as ASC, and then ASC further recruit caspase-1 to form an inflammasome. And these inflammasomes are pretty interesting in the cell because they exhibit as these fairly large conglomerate of proteins, and oftentimes it's a single entity in the cell in the micrometer scale. And it is within these molecular assemblies that caspase-1 is activated. And then caspase-1 has a few preferred substrate. So we already know it cleaves pro-IL-1 to generate the mature form. Actually cleaves several members of the IL-1 family, also including interleukin-18. At the same time, caspase-1 or other so-called inflammatory caspases also cleave gasdermin D. This is a protein that was realized in 2015 as a substrate for inflammatory caspases. And the cleavage of gasdermin D leads to the release of the N-terminal fragment for its pore formation activity on the cell membrane or other organelle membrane. And this induces the release of the processed cytokines. And then in severe cases, also the lytic form of cell death that is highly inflammatory, known as pyroptosis. So here I marked several of the proteins that has been the target for therapeutic efforts in the field, pretty much almost every protein in this pathway. So most of the disease relevance, if you will, of inflammasomes really came from studies on NLRP3. So NLRP3, as it turns out, senses many different things, and perhaps the overarching principle that it senses is some kind of cell membrane damage. And the cell membrane damage induces some kind of potassium efflux, and that indirectly activate NLRP3. So here I show you some examples of stimuli that can activate NLRP3. bacterial toxins that generate cell membrane damage, ATP receptor, which activate a potassium channel, or any other way to damage a membrane. And I really like this family of stimuli. These are particularly structures that can be endocytosed and then damage the endosome. And that leads to potassium efflux and NLRP3 activation. And there are many kinds of abnormal aggregates in the organism, in the human physiology: uric acid crystals, cholesterol crystals, amyloids, asbestos, just to name a few. So I'd like to show that NLRP3 has many disease mutations. Although these are very rare hereditary inflammatory disorder, but these mutations that hyperactivate NLRP3 causes multi-organ inflammation. And I think this is the first hint that NLRP3 activation may have a far-reaching effects in different organ systems and in different disease settings. And this came to this conclusion that inflammasome is at the center of many metabolic, cardiovascular, neurodegenerative diseases. And I think the challenge for the field is really to figure out which subset of these diseases are completely driven by inflammasome activation. So the NLRP3 or other NLR inflammasome often have these three different proteins in its assembly. And we've known for some time that it is the PYD-PYD interaction and the card-card interaction. These are fairly small domains that can form filamentous structures, such as these that I'm showing you here, that forms the scaffolding for the caspase domains to be brought into proximity for their activation. And we have, we and others in the field have shown that these signaling clusters in the cell is a conglomerate of filamentous structures, and we can recapitulate that using in vitro reconstitution. So we know a lot about this downstream caspase activation process, but we know very little about how these NLR proteins that has a nucleotide binding domain, they're really ATPases that mediates oligomerization. Very little is known about how these proteins domains in inflammasomes exert their functions. So the only example in the case of inflammasome was studies that my lab and other labs performed a few years ago on this particular inflammasome. It's called NAPE, NLRC4 inflammasome. NAPE is another member of the NLR family. It's also known as NLRB, but NAPE is a more recognized name for NLRB. In any case, for the case of NAPE, upon recognition of flagellin, so these are flagellin that gets injected into the cytosol. Upon recognition of flagellin, the NAPE protein is activated by a conformational change in the ATPase domain. And then this protein is able to polymerize the NLRC4 protein and successively cause its conformational change from this closed state to this more open state, which is ATP form and ADP form here, to form this platform. And then the small domain, the helical assembly domains in the center can then mediate the assembly of the inflammasome. So what is the situation for NLRP3? So NLRP3 is a little bit more complex because there are other proteins that are essential for NLRP3 activation. So NEX7 was shown to be important for NLRP3 activation. And if you knock out NEX7, you don't get NLRP3 activation, at least in mice. And the kinase activity is not required. K means it's a kinase. The kinase activity is not required, suggesting that NEX7 really act as a scaffold to promote NLRP3 activation. So interestingly, NEX7 is a centrosomal kinase. So as I will show you later, inflammasomes are also localized at the centrosome when they are formed. So NEX7 is a centrosomal kinase and it's important for mitosis. And because of this relationship, it is known that inflammasome activation does not occur in mitotic cell because of this competition with NEX7. And by contrast, mitotic cells cannot have inflammasome activation. So because of the importance of this protein in NLRP3 activation, a few years ago, we used a NLRP3 construct that doesn't have the PYD because we worried that the PYD would cause a lot of aggregation and make structure studies not possible. So we solved the cryo-EM structure of NLRP3 delta PYD in complex with NEX7. So this structure, of course, revealed the mode of interaction between these two proteins. But probably more importantly, I think, is that it gives you a framework to understand how NLRP3 NEX7 inflammasome can be assembled. So this is a model in which we generate a conformational change in the ATPase domain to induce its active conformation. And then we model this onto the NLRC4 inflammasome. What's interesting is that in this model, the NEX7 protein is bridging two leucine-rich repeats from the NLRP3 protein, suggesting that maybe the role of NEX7 is to bridge NLRP3 molecules in the assembled NLRP3 and therefore promote the assembly. But is this all the story? So when Ludmila joined my lab a few years ago, she decided to look at full-length NLRP3 with the PYD domain. So she was able to express the protein from HEC293 T cells. Actually, she grew a lot of plates. And what's interesting is that she found that some NLRP3 that she purifies are very large, as shown on this sucrose gradient. And when she looked at these large structures, she saw that they form these ball-like things, fairly large and ball-like, large, probably around 25 to 30 nanometer in diameter. And she was able to use nucleotide to stabilize these structures that we later called cage, and solve this structure to a sufficient resolution to be traced. So the structure itself is quite interesting. It has 2 layers. It's a dodecamer. It has 6 molecules on the top and 6 molecules at the bottom. And if you look at how this cage is assembled, it doesn't assemble through the NAC domain or the ATPase domain. And in most cases, the active conformation or the active state of the NLR proteins assemble into oligomers through the NAC domain. And this is not the case for this double ring cage. So this was already telling us the structure may be still in an inactive state. And this is consistent with the fact that we didn't really stimulate the cell to activate NLRP3. So instead of using the ATPase domain, it's the leucine-rich repeat domain that is used to mediate the assembly between the 2 layers in the cage. And in the middle here, the density has fairly low resolution, but we think it's the N-terminal PYD domain that has already formed into filaments, but they're hiding inside the cage structure. So we've known previously that the filamentous assembly by PYD is a unidirectional process. And because we saw that the cage structure is not able to nucleate the filament formation of the next protein in the cascade, so it cannot recruit ASC, we hypothesized that these PYD filaments actually faces inside, or the polymerization interfaces faces inside, so that it cannot nucleate, or ASC cannot see this interface and cannot be induced to polymerize. So again, this confirms that the cage structure is an inactive structure. So what was a bit surprising was that during the protein purification process, Ludmila noticed that most of the proteins in her prep actually belong to the membrane fraction. And even though her initial prep came from the cytosolic fraction, she could also take the membrane fraction, solubilize the membrane with detergent, and then reveal the same kind of cage-like structure. Also in macrophage, these are cells that can activate the inflammasome. In macrophages, again, most of the NLRP3 are in the membrane fraction. And when she ran a native page to look at what kind of structure that may be that may exhibit in these membrane fractions, she saw that the main band in the membrane fraction is exactly at the same migration position of purified NLRP3 cage, suggesting that indeed, even in macrophages, the main membrane fraction contains these cage-like structures. This was surprising in a way because a few years ago, James Chan's group have shown that NLRP3 is diffusely distributed before activation, but after activation, this is nitrorising, this is the bacterial toxin, it then gets recruited to the trans-Golgi network. So you may know that the trans-Golgi network is enriched in phosphatidylinositol phosphates. So there are lots of PIPs on these membranes. As I will show you that in macrophages, so these are done in HeLa cells with transfected NLRP3, but in macrophages, NLRP3 is already localized to the trans-Golgi network, which is this region of the cell before stimulation. So, this recruitment to the trans-Golgi network and the dispersion of the trans-Golgi network into these speckles is really important because we have shown a few years ago that the NLRP3 inflammasome are assembled at the microtubule organizing center. And if you look at what's on the MTOC, you have not only NLRP3, but you also have the TGN marker, TGN38, suggesting that it is the vesicles, the small vesicles that gets dispersed by inflammasome activation that is transported by the microtubule to the MTOC for NLRP3 inflammasome activation. So in order to look at how does the cage structure binds to the Golgi, we looked at these so-called polybasic region. So these regions are disordered in the structure, but they should have been presented at the outside part of the cage. So this region is between the N-terminal PYD and the ATPase domain, and they are enriched in positively charged residues. So the cage itself has very strong affinity to these PIPS lipids in vitro. And if you express a NLRP3 construct that has these residues mutated, you see that they no longer form cage structure. But if you take the linker mutant, but add a domain that can traffic the protein to the Golgi, now you see the formation of the same or similar cage-like structure as in the wild type. So that suggests that this Golgi binding is important for the formation of the cage. So here I show you in macrophages, these are just so-called primed by LPS. What LPS does is to activate toll-like receptor 4 and increase the expression of NLRP3, but before inflammasome activation. So as you can see here, NLRP3 is already co-localizing with the TGM marker before activation. And after activation, it disperse the trans-Golgi network and form the single puncta that's localizing on the mTOR. And this Golgi dispersion effect is gone if you don't have NLRP3, no NLRP3. But if you reconstitute a NLRP3 minus macrophage with the wild type, it goes back to, I thought I have a slide on that. It goes back to the same phenotype as the wild type, but if you reconstitute with the mutant, then you don't get this phenotype, and suggesting that the cage not only localized to the trans-Golgi network, but also is important for generating the vesicles of Golgi vesicles. And if you look at LDH release as a surrogate of cell death, the cage defective mutants also do not induce cell death. So this led us to the conclusion of our study, that is no cage, then no vesicle, then no transport to the mTOR, and therefore no inflammasome activation. So we don't really know how this NLRP3 cage structure is important for the dispersion of the trans-Golgi network, but we hypothesize that perhaps because there are multiple membrane binding elements in the cage, at least two sides, and perhaps because of that, it can help to deform the membrane, and perhaps that can be an initiating factor or promoting factor in the dispersion of the trans-Golgi network. This is something that we are interested to test in our future work. So now I showed you previously that NLRP3 containing vesicles would transport to the mTOR, but why? So one potential reason is that NEX7, the centrosomal kinase, is mainly localized at the mTOR. And if you remember the structure of NLRP3 in complex with NEX7, this binding surface is exactly overlap with the cage formation interface. This is the two leucine-rich repeat that mediate the cage interaction. And therefore, one would expect that NEX7 would compete with the cage and maybe even disrupt the cage when the cage structure arrives at the mTOR through the microtubule transport. So in vitro, indeed, when we mix the cage structure with NEX7, you see the generation of smaller complexes and therefore the breakage of the cage. So these then led to our hypothesis on the role of trafficking and concomitant conformational change, the role of trafficking and conformational change in the NLRP3 activation pathway. So at the trans-Golgi network, you may have an equilibrium between the monomer and the cage is preferentially formed on the membrane. You need a membrane, it looks like you need a membrane scaffold to form the cage. And then upon stimulation of an inflammasome, then Golgi dispersion occurs, and then these vesicles get transported to the mTOR. And at the mTOR, the NEX7 may then simply peel off the two layer of the cage, because the PYD domain are still associated together, so that the two layers are likely still associated with each other. But you open up the 2 layers, and therefore then NEX7 and perhaps also additional NLRP3 molecules may be assembled onto it, and this then form the central platform, central PID platform for the recruitment of ASC and then caspase-1 polymerization. There are many different gaps in this hypothesis, and we're interested to elucidate them in our future studies. So as a summary, we've shown, at least for NLRP3, the activation mechanism is not at all straightforward. It requires a lot of different steps and regulation, and perhaps this is related to its need to sense some kind of disturbance, and maybe these steps are required for this sensing process, this indirect sensing process. So I didn't talk about this. I just want to show you that the same family of protein, NLRP6 and NLRP1, turned out to use completely different mechanism for their activation. In the case of NLRP6, a step of phase separation is required before the assembly of the filamentous structure within the phase separated entity. And for NLRP1, a cleavage process mediated by pathogen or other stimuli is required to allow the release of the C-terminal fragment in order to form the inflammasome platform. And then they all converge to the common downstream bifilament formation. So to me, it's pretty remarkable that the same kind of proteins can perform different function depending on the circumstance. And it is perhaps an ultimate example of structure serving the function. So for those of you who are not in the field of structural biology, I just want to point out that we are also evolving. In fact, some of the inflammasome structures we solved, we used AlphaFold to help the model building. And this is a ultimate example of using AlphaFold to build medium resolution structures for essentially all the subunits in this nuclear pore complex map. So with that, I want to thank all the fantastic postdoc and students from my lab who made the work possible. Many of my trainees are of the Chinese origin, and I want to thank all of them for their hard work and creativity, and I thank you for your attention.
So we have time for a few questions. If you are in the panel, you can directly raise your hands. For others, you can type the question in the Q&A. So I have a question. So did you saw the density of ATP or DATP in the keyed structure of NLRP3?
No, the resolution was not good enough, but the confirmation clearly tell us that it is an inactive conformation. It should be the ADP form. Oh, okay. So maybe the reason you're asking is because we used DATP. So we use DATP to stabilize the structure. I'm going to stop sharing, okay. Oh, I lost my control. I don't know where's the, can you stop sharing for me or no?
That's how, if you want to, oh yeah.
Yeah, is it stopped? Yeah, okay, I couldn't find the button. Yeah, so we use DATP. It just something that she tried, and it turned out it's the best one. And even though NLRP3 can hydrolyze ATP, it can also hydrolyze DATP. So she has data to show that. So we think what's in the structure is most likely DADP. And this is quite similar to the case for apoptosis. There is the D version, the deoxy version that has better affinity to the catalytic center of these proteins. Okay,
so if there's no further question,
must be crystal clear. Yeah.
Yeah, there's a question. So for the double linkage structure of NLRP3, how conserved is this region in NLRP3 from other species?
Very conserved. Yeah, so this is definitely conserved. This is actually also in the paper. So we think this is a feature that's shared certainly between NLRP3 in different species. Although there are so there are cases in which the leucine-rich repeat actually so BAT has a NLRP3 that doesn't have the leucine-rich repeat, and those and NLRP3 is highly suppressed, in fact, in BAT species. So there is a correlation between the function of the K structure and how it functions in different organisms.
So without a further question, now let's move to our second speaker in this session, Professor Eric Gao, who actually has been a hero to me in membrane protein structure biology since I was graduate student. Eric received his bachelor degree and a PhD degree from Harvard University.
I'm very excited to share with you an interesting story, emerging enterococcus performing toxin with MHC class 1 as receptors. Enterococcus is gram-positive bacteria and is extremely widespread. This figure shows the diversity of hosts of enterococcus. We can see a wide distribution of enterococcus in birds, insects, mammals, including land animals and aquatic animals. Enterococcus is gut microbiota of human and animals. It's a dangerous pathogen. In healthy people, enterococcus does not usually cause a problem, but if it spread to other areas of the body, including wounds, blood, or urine, it may cause life-threatening infection. People in hospital or who have underlying health conditions are at higher risk of developing a deadly infection. So enterococcus can cause infective endocarditis, account for 30% of IE case worldwide with 25 mortality rates, and it can cause UTI that account for 30% of UTI case among 150 million patients. It's also responsible for 10% of bloodstream infection, up to 28% mortality rate. So it's dangerous pathogen. Among the enterococcus species, the most dangerous are the E. faecalis and E. faecium because they are multi-drug resistant. Those are hospital-acquired infection and two common bacteria isolated from clinical case. The E. hyrium is the most abundant enterococcus species in human microbiota. Recently, a paper shows it can regulate immune response to tumor antigen. Though being potent pathogens, previously no toxin of enterococcus targeting to human has been identified. We have continuously collected, sequenced, and analyzed the enterococcus strains since 10 years ago to understand the pathogenesis of enterococcus. By genome sequencing and bioinformatic analysis, we found a potent performing toxin gene in E. faecalis, E. hyrium, and E. faecium. We term them as EPEX 1 to 8. There are growing numbers of homologs of EPEX gene in E strains deposited within the last few years. This figure shows the global geographical distribution and a source of isolation for all epochs carrying enterococcus strains. The epochs genes are widely spread in the world, collected from 7 continents. These strains were reported from diverse sample sources, like food animals, including chicken meat, turkey, dairy cow, wild animals, environment, including tap water and soil, and people samples, including stool and human intestine. So they are very close to our daily life. Bioinformatic analysis shows the epochs family are very close to the delta toxin, alpha hemolysin, and NADB. Those 3 protein performing toxins. So we suspected that the epochs are performing toxin since it share high identity with delta toxin, NADB, and alpha hemolysin. Performing toxin present a broad class of virulent proteins secreted by pathogens. It can disrupt the target cell membrane, which they form pores on, inducing cell damage and death. The primary member of performing toxin are alpha performing toxin and beta performing toxin, which features pore consist of alpha helix and beta barrel. They are produced as soluble monomers that can oligomerize and form transmembrane pores on the cell membrane. The hemolysin family is an important branch of beta performing toxin. Alpha hemolysin, also known as HLA, is a well-studied hemolysin family member. It's produced as 300 residue monomer and assembles into a mushroom-shaped hemeric pores. HLA can lysis the red blood cell and suppress the immune system. So the bacterial performing toxin are widely implicated in bacterial infections and essential for pathogenicity. It can disrupt epithelial barriers, disable immune cell, and damage tissues. It's critical for understanding pathogenesis and therapeutic development. So to validate the epochs are performing toxin, we produced and purified the epochs 1 to 4 protein and added them to the HEK2 lysolytic cell. All 4 epochs can induce cell death of 2 lysolytic cell. 2 lysolytic cells were rounded and died in 10 minutes after epochs treatment, whereas the cells were survived after being treated the heat denatured epochs. We then assess the susceptibility of HeLa cells by exposing cell to serial dilution of toxin and measuring cell viability. We found that the EPAX2 is one of most potent performing toxin known for HeLa cells, and is toxic 100-fold than other EPAX. To further confirm EPAX cytotoxicity is because of performing toxin activity, we performed the liposome leakage assay. We detected the fluorescence dye signal in solution when the liposome incubated with EPAX, suggesting that the fluorescence dye released from liposome. This result shows that all 4 EPAX are capable of inducing lysis liposome in vitro. Furthermore, we directly observed the EPAX2 pore on liposome by negative staining electron microscopy. To see is to believe, very likely, we solved the structure of EPAX1 and EPAX4 by crystallography and cryo-EM, demonstrated the EPAX are performing toxin. They contains a cap domain formed by core beta sandwich region and a rim domain with several aromatic residues that likely contact the cell membrane, and a stem domain forming transmembrane beta barrel pores. The EPAX overall architecture are similar to alpha hemolysin. However, the octameric configuration of EPAX4 pore is unique, since single component of beta barrel performing toxin previously reported all from hemeric pores. Another unexpected feature of the EPEX4 pore is the formation of a second beta barrel that sits on the top of the cap region. The diameter of this top domain is similar to that of the transmembrane beta barrel pore. The top domain, the cap domain, and the beta barrel pore form a continuous channel. These unique features make EPEX a new branch of beta-performing toxin. So as we characterize the EPEX as cytotoxic performing toxin, next we further investigate the host receptor for EPEX. Since EPEX is the most potent toxin in EPEX families, we next focus on identification of receptors for EPEX2 using genome-wide CRISPR-Cas9 screen. A genome-wide single-guide RNA library was transduced in HeLa cells that stably express Cas9. Cells were selected with increasing concentration of EPEX2. The surviving cell were harvested and sent to do next generation sequencing. The bioinformatic analysis result shows that the top hit is B2M. B2M binds to HLA to form MHC class 1 complex. The MHC class 1 is a cell surface protein essential for acquired immune system to recognize the foreign molecules. HLA also identified in our screen. Binding of B2M is critical for proper trafficking of MHC class 1 onto cell surface. Thus, knockout B2M would block cell surface expression of all MHC class 1 complex. So to validate the top hit, we generated stable B2M knockout HeLa cells. B2M knockout cells showed resistance to EPEX2 compared with wild-type HeLa cells. Pull down assay result shows EPEX2 can pull down MHC class 1 complex, but not B2M or HLA protein only. To assess the potential species selectivity, we performed the pull down assay using GST tag the EPEX2 pull down against the MHC class 1 from human, equine, murine, bovine, and porcine. The result shows the EPEX2 prefer to recognize human MHC class 1 and is homolog in equine, bovine, porcine, but not in murine. So next, we want to explore the role of EPEX in Enterococcus pathogenesis. Since the immune suppression is a major function of many performing toxins, so we performed co-culture experiment that co-culture EPEX2 carrying strain. The name is DIV0147 with freshly isolated PBMCs. And another Ephysium strain use a control. This control strain share 99% DNA sequence identity, but not carrying the EPEX2 genes. The damage of the PBMC were measured by LDH release assay. LDH is a soluble cytoplasmic enzyme that is present in almost cells and is released into extracellular space when the plasma membrane is damaged. The result shows the PBMCs were damaged after co-culture with EPEX2 carrying strain, whereas the control strain showed no toxicity. Adding a EPEX2 antibody can neutralize the cytotoxicity of the EPEX2 carrying strain. Since the disruption of epithelial barrier is another common function of performing toxin, we co-culture the human intestine organoid as monolayer in transwell. And then we carry out co-culture experiment by adding EPEX2 carrying strain into the upper layer of the transwell. Co-culture 8 hours, we can detect the damage of intestinal organoids monolayers as measured by LDH release assay. Meanwhile, by adding dye into the upper layer of the transwell, we also detected the disrupted cell-to-cell junction by increasing leakage of dye from upper layer into the lower layer. But we didn't detect any damage in the control strain. Adding the EPEX2 antibody can neutralize the EPAX2 carrying strain toxicity completely. So this result indicates the EPAX2 play a critical role in Enterococcus pathogenesis. To summarize the talk, I hope I have convinced you that we identified a family of Enterococcus performing toxin, EPAX, in Enterococcus strain isolated across the globe. Structurally study revealed that EPAX form a new branch of beta barrel performing toxin with a unique top domain. Through a genome-wide CRISPR-Cas9 screen, we identified MHC class 1 as a receptor for EPAX2. EPAX2, which that is preferred to recognize human MHC class 1 and the homologue of MHC class 1 of equine, bovine, porcine, but not murine one. Co-culture with EPAX2 harboring Enterococcus strain can damage human PBMCs and intestinal organoid, and this toxicity can neutralize by EPAX2 antibody, demonstrating the toxin-mediated virulence of EPAX-carrying Enterococcus. There are also some interesting points to speculate. The first, the EPAX2 are potent emerging toxins located in plasmid. So it could be very likely that the EPAX gene can acquired by bacteria and turning the harmless bacteria into dangerous pathogen. And pathogens often evolve and emerge from natural reservoir outside of humans. For example, the COVID-19 virus, and recently the monkeypox jumped to human from animals. So EPAX2 can recognize the MHC class 1 complex in humans and a major farm animal, which is very close to our daily life. So starting the EPAX emerging toxin will call people attention because it's a new potential threat to human health. So I would like to acknowledge my mentor, Dr. Ming Dong, for his strong support and help for my project, and my colleague, especially Dr. Song Haitian. He helped a lot in identifying the EPAX receptor by genome-wide CRISPR-Cas9 screen. And Pan Yang from Jonathan Branham lab, he helped solve the CRO-EM structure of EPAX1. And Franciva from Michael Gilmore's lab, he is a bioinformatics guy and initiated this project. And Tianming Fu and Jianping Ran from Hao Wu's lab helped a lot for crystal structure study. I really appreciate your kind support and help. Thank you so much for your listening and attending.
Hey, Xiaozhi, really great talk. I do have one question on my end.
Sure.
So the binding of the toxin to the MHC molecules, does it block its function, or is it downregulates its expression? Because I'm curious to know because when you know cells lose expression of MHC1, it might make them more sensitive to killing by natural killer cells. I wonder if you guys ever look at that or consider looking at that aspect.
They binding the MHC class 1. That is MHC class 1 is a target of it's a receptor target of the EPAX2. If we block the MHC class 1 or knock out the MHC class 1, the toxin lose its toxicity on the cell.
Okay. I think I can also ask another question
Sure.
on the behalf of Long from the panelist. He's asking, have you looked into the cell survival strategy? Does escort rescue the cell leakage?
No, we haven't studied the cell survival strategy.
Okay, great. I think for the sake of time, we'll have to move on to the closing of today's symposium. Thank you so much, Xiaozhi, for the presentation. I'm going to spend the next, thank you. I'm going to spend the next few seconds addressing the closing remark for today and also make some acknowledgements. First, I would really like to thank all the guests of today's symposium, including the opening remarks speaker, session speakers, as well as session moderators, especially those of us who join us from, despite the difference in time zones. It's really awesome scientific discussions and communications. Thank you to all the audience for registering and attending today's symposium. And I would like to take the next few minutes to acknowledge a few more people. First of all, the HMS-CSSA team, including a lot of my colleagues. We're all volunteers who are postdocs, mostly postdocs around the community, and also the faculty advisors of HMS-CSSA, as well as reviewers for this year's HCLS Distinguished Research Award, who are also a lot of faculty members around here. Thank you to our sponsors, including Biocytogen, BGI Americas, and Cowen Biosciences. And a big shout out to the organizing committee members for the 2022 HCLS. We're all members of the HMS-CSSA, and big thanks to E as today's host. She did an awesome job. And also a lot of my colleagues who are responsible for inviting speakers, organizing the sessions, advertising the events, as well as physically being here in the conference room with me today for this event. There's just too many people to thank, and I would like to ask all the audience to keep supporting the work of HMS-CSSA, and if any of you guys are interested in sponsoring us in any way, feel free to contact us. We have our WeChat official account. Please follow us. And also, if you have any questions, don't hesitate to contact us through the email. And thank you again. And I think this is it for today's symposium, and have a good weekend, everybody.